Good morning, ladies and gentlemen, and distinguished invitees. Welcome to the Symposium on Indo-Sri Lanka Collaborative Research Program. This event is organized under the scientific cooperation between the Government of Republic of India and the Government of the Democratic Socialist Republic of Sri Lanka. Historically, relations between Sri Lanka and India were built on shared cultural and religious heritage over the course of several millennia. In more recent times, relations between the two countries have extended over many areas, including scientific research, the results of which we are about to witness today. To commence the inauguration ceremony, let me invite the Director General of the National Science Foundation, Dr. Sepalika Sudasinghe, to deliver the welcome address and the opening remarks.
Thank you. Good morning. Greetings from National Science Foundation. So, Secretary to the Ministry of Education, First Secretary Education to the High Commission of India in Sri Lanka, additional secretaries, heads of institutes, research teams, distinguished invitees, additional directors, heads, and other staff from NSF, and also specifically the researchers here joined from India and Sri Lanka. Ladies and gentlemen, it's indeed my pleasure to deliver the welcome address of this important symposium on Indo-Sri Lanka Collaborative Research Program. On behalf of the Chairman, National Science Foundation, Board of Management and staff of the National Science Foundation, I wish to extend my very warm welcome to Mr. M.N. Ranasinghe, Secretary to the Ministry of Education, and Mr. Vivek Gupta, First Secretary Education in the High Commission of India in Sri Lanka for gracing this event. The additional secretaries, heads of institutes and staff of the Research and Innovation Division of the Ministry of Education who had initiated and have been collaborating from the beginning of this program are warmly welcomed. We most welcome Professor Ajit De Silva, Dr. Mubarak, and Professor Tantirigoda for agreeing to moderate the technical sessions. Heartfelt welcome goes to you, the researchers, and your teams for your excellent work that we will be presented at the symposium. Ladies and gentlemen, your presence here, your participation in this event is much also appreciated. So if I talked about in brief the background of this program and also about NSF, NSF is a state-funded institute which is at present comes under the Ministry of Education, and we committed to generate knowledge and also dissemination of research findings and transfer of knowledge, and most importantly, to ensure the effective utilization of knowledge for the greater benefit of the people of Sri Lanka. We support and embraces research in all fields of fundamental, applied social sciences as well. The NSF support R&D activities at all fields of S&T, nationally as well as with international collaborations, of which the outputs and outcomes ultimately benefit to the general public. The first program of this cooperation between two countries was signed on the 22nd of September 2011. Accordingly, both parties had agreed to execute science and technology based on projects and workshops. Today's symposium is the result of this signed agreement in 2017, where research teams from India and Sri Lanka have collaboratively worked on these themes of the projects under nationally important research areas for both the countries. We are pleased to note that from 2017 research cycle, as 17 projects have been successfully completed, and by now 28 journal publications, 211 conference papers, and 21 abstracts have been published by the research teams. Scientists and researchers from both countries have benefited from these projects through sharing knowledge and research experiences. The exchange programs have been exposed young scientists not only to new technologies, but also to the research cultures of both countries. This program has helped in capacity building as well as engage scientific cooperation between many Indian and Sri Lankan universities, research institutes, and other science and technology institutes. We hope to come up with suggestions for way forward during the discussion to sustain the achieved success as well as to initiate and execute the program. I sincerely hope that we would have a productive, successful symposium today, resulting in positive recommendations. Thank you very much.
Thank you, Madam. And now, ladies and gentlemen, we will be launching the booklet showcasing the research highlights of the Indo-Sri Lanka Collaborative Research Program. You can access the booklet by scanning the QR code given at the end of the program document. And also, I'm sure all of you got a CD containing the soft copy of the booklet. We can now witness the research highlights booklet on the screen. Moving on in the agenda, I'm pleased to invite the Secretary to the Ministry of Education, Mr. M.N. Ranasinghe, to address the audience.
A very good morning to you all. Mr. Vivek Gupta, the First Secretary of Indian High Commission in Sri Lanka, Additional Secretary, Director Generals, scientists, researchers, ladies and gentlemen. First of all, let me thank the National Science Foundation and the Research and Universities of the Ministry for having invited me to grace this very important occasion. I feel honored and consider it a privilege to address you at this Indo-Sri Lanka Second Symposium Research Program. Since independence, Sri Lanka and India have shared well-established diplomatic relations, sharing a land and maritime border. We are connected through important economic, diplomatic, cultural, and other ties dating back 2,500 or more years. Bilateral relations between the two countries have shaped through a number of key agreements in different economic, political, and social environments. This outcome is one such cooperation between Sri Lanka and India for collaborative research, advanced research. This program has provided a strong force for bolstering research ties and knowledge sharing between our two countries. The benefits gained by our country from this program are immense. We benefited from the research strength of India, which is vast. With support from the government of India, considerable investment and development has occurred in different sectors such as agriculture, healthcare, space research, and nuclear power through scientific research in India. Our scientists and researchers have benefited from these developments, sharing the knowledge and research experience with the Indian counterpart, and there were many exchanges between the two countries. The exchange programs built in the two programs have exposed our young scientists not only to the new technologies, but also to the research culture in India, learning good practices and effective methods and procedures. I hope the Sri Lankan scientists also would contribute in a similar manner in enhancing research culture in India. We all know research publication in turn elevate the research and innovation status of the country. All these activities I talked about lead towards increasing both our country's knowledge and technology outputs, and in turn affecting the status of our country in the Global Innovation Index. We should not forget the various workshops supported by this program. Although less in number, their importance and significance is not to be taken lightly. Workshops help to disseminate the findings, knowledge gained, and the conclusions made from the research carried out to the public in a very effective manner. They can be developed to support public engagement at any stage of a project. The flexible nature of workshops means that they can be exploratory as well as designed to test and communicate research outcomes. Experts and integrated members of the public can be brought together in workshops to explore research issues. It is also possible to reach those who do not read research journals in general, but interested in the subject matter, so that their ideas can be captured to validate the results. I have no doubt that the current round of projects and workshops will be successful and benefit both our countries. With the global outbreak of the coronavirus, every country is facing a multitude of economic challenges. Therefore, we must ensure that the monies are well spent funding projects with socio-economic impacts to our countries, where benefits can be reaped short term. In every field of science and technology, together, we must ensure that allocated monies are spent to find new technologies, explore new frontiers, and find new applications of existing technologies. The collaboration started and connections made during these projects do not stop here. They will become stronger and expand further, building friendship and other alliances, and going further afield, building multinational networks and creating an enabling environment for sustained engagement in mutually beneficial areas. Together, we can become strong, excelling in research and innovation, and work towards finding solutions to national and global problems. So the Ministry of Education in this context have identified this role very clearly. Unlike in the past, now the Ministry of Education has taken a very strong stance on developing the general education sector, vocational and technical education sector, higher education sector, plus science and technology sector. So now everything is in a single basket where symbiotic relationships can be built to ensure that constructive and integrated approaches can be put into practice. So we, Honorable Minister of Education, is well aware that the science and technology can play a very, very significant role in putting the country into the real and practical mode when it comes to addressing the development challenges. So we have very clearly identified that during the past couple of decades, forgetting the science and technology, we have achieved this status. We have not taken seriously the research outcomes, research efforts, and encouraging research. We have not done sufficient enough in the recent past and also past. Therefore, under whatever circumstances, we must get together and give highest priority to the research and development sector. So therefore, it is not sufficient to undertake research and then share findings and keep it in a shell. So we need to go for a formula which should be N to N plus plus. Or in other words, A to Z plus plus. Plus plus means this is a round. This goes again, comes back to the original point. When it comes to research findings, we have to put into practice in the general and the practical domain. So there again, you will find further opportunities and further gaps when it comes to practical implementation of the research findings. So therefore, we are, as the government, we have identified three basic roles that should be played. First and foremost is the guiding the government through the government policy mechanism. We must guide the institutional framework that undertake the development efforts. So the research and development is a very significant area in that context. Therefore, the government has identified the research and development area as a priority when it comes to introducing new policies. And the second aspect is the facilitation. The government must facilitate through the budget mechanism or fiscal administration how we can put the research and development platform into that appropriate level. So therefore, when it comes to mobilizing resources through the government or through bilateral or multilateral arrangements, we will give priorities to this area. And the third and a significant aspect is the regulatory part. We need to work together. There are a couple of similar agencies in the field that are working in isolation, which will not produce results, which will not in any way produce synergies, and the strength will be marginalized. So therefore, we as the Ministry of Education will support under these three key major areas to the institutional framework, which is really involved in this all efforts. So therefore, finally, certainly the successful completion of a research project involves communicating research findings to keep the society informed. It will add value to the information derived from the research. Therefore, traditionally, peer-reviewed publication and presentation in conferences are the two most common dissemination practices. There is no way to reach the general public through them. Conducting a symposium with our institutional heads and other relevant stakeholders may ensure understanding and utilization of research findings into practice. Therefore, finally, I again reiterate the significance and importance of the government of India playing in this crucial situation. The government of India has come forward immediately after the COVID pandemic and the subsequent economic downturn to help us. So we definitely are thankful to the government of India and especially the Indian High Commission in Colombo for their continued support to build a stronger relationship between the two countries. Therefore, I extend my sincere gratitude to all the researchers and wish you every success in this very important endeavor. Thank you very much.
Thank you, sir. Ladies and gentlemen, it is the time to appreciate the efforts taken by the research teams to deliver high-caliber research output. In order to do that, we will be presenting certificates to all the research teams. One member from each team can come up here to receive the certificates for their team. And to do the honors of presenting the certificates, I am pleased to invite Mr. M.N. Ranasinghe, Secretary to the Ministry of Education, and Mr. Vivek Gupta, First Secretary, Education of the High Commission of India to Sri Lanka. And to accompany them, Mrs. Nadika Vataliyada, Additional Secretary, Development and Innovation of the Ministry of Education. The first project under the Indo-Sri Lanka Collaborative Research Program studies in surface sterilization of spices using non-thermal processes. And the principal investigator is Dr. P.N.R.J. Amunugoda. And the co-PI is Ms. R.C. Pitipanarachi. The team member is Mr. W.A. Buddhika Sampath Veerasinghe and the research student, Mrs. A.B.G. C.J. De Silva. The next research title, High Throughput Genotyping to Expedite the Genetic Characterization and Dissection of Important Agronomic Traits of Tea. The PI is Professor R.N. Attanayaka, and the co-PIs are Mr. J.D. Kottavarachi, Mr. M.A.B. Ranatunga, Dr. A.M.T. Amarakon, and Dr. M.T.K. Gunasekara. The next project is Biopolymers Biosurfactants Based Delivery Micro Nano Vehicles for Safe Delivery of Food Bioactives and Nutraceuticals. And the principal investigator is Professor Nedra Karunaratna, and the co-PI is Dr. K.M. G.K. Pamunuwa, and the research student is Ms. I.F. Shakur. The next project, renewable chemicals from the plant of Indian subcontinent, isolation, self-assembly, and utilizations in drug delivery, medicine, and catalysis. The principal investigator is Dr. M.T. Napagoda, and the co-principal investigator is Ms. K.D. Vijayasekara, and the research students are Ms. E.K.S. Niranga and Ms. P.D.U. Pavitra. The next certificate is for the project titled Bioprospecting Endolycanic Fungi for Mangroves in Negombo Lagoon in Sri Lanka and Gulf of Khambat, Gulf of Kutch from Gujarat, India, an untapped treasure trove for discovery of special structures and bioactive compounds. And the principal investigator is Professor P.A. Paranagama, and the co-principal investigator is Professor R.N. Athanayaka, and the research students are Ms. R. Veerasinghe and Ms. N. Pereira. The next project, in vitro and in vivo epigenetic investigations on anti-cancer and radiopriming effects of different parts of Mangifera indica, India, and Mangifera zelenica, Sri Lanka, and impact of green-silver nanocomposites. And the principal investigator is Professor K.H. Thennakoon. And the co-investigators are Professor S.R. Samarakon and Dr. M. Edirivira. And the research student is Mr. A.A.D. Nirmal Pereira. The next certificate goes to Dynamic Analysis and Shape Control of Inflatable Structures for Space Application. And the principal investigator is Dr. H.M.Y.C. Mallikarachi. The next project is titled Wireless Sensor in Network Distributed Detection Mechanisms for Structural Health Monitoring in Civil Structures. And the principal investigator is Professor P.D.R. Disanayaka. And the co-principal investigators are Dr. A.J. Dhammika and Dr. K.K. Vijayasundara. The next project, Design and Development of Detection and Extinguishing Systems for Forest Fire using Sensor Networks, Aerial and Ground Robots. And the principal investigator is Dr. K.V.D.S. Chaturanga, and the co-principal investigator is Dr. S.W.H.M.T.D. Lalitharatna. They are not present here. They will be joining online only. The next project is Reconfigurable Architecture for Solar Photovoltaic Microgrid Systems. And the principal investigator is Professor K.T.M.U. Hemapala. And the co-principal investigator is Dr. W.T. Prasad. And the research students are Ms. K.A.H. Lakshika and Mr. M.A.K.S. Boralasa. The next project, Design and Development of Hybrid Wind Solar Power Generation System using multi-level inverter for grid-connected application. And the principal investigator is Dr. R.P.S. Chandrasena. And I'm afraid he is also not with us here today. And the next project is Electronic and Photovoltaic Properties of Two-Dimensional Hybrid Organic and Inorganic Perovskites. And the principal investigator is Dr. M.L.C. Artigala, and the research students are Mr. N.L. Adihetti, Mr. D.R. Ratnasinghe, and Mr. P.M.W.P. Sampath. The next project is titled Forming of Automotive Materials at Elevated Temperatures and Selection of Lubricants for Sustainable Manufacturing. The principal investigator is Dr. P. Gamage, and the co-principal investigator is Dr. N.K.B.M.P. Nanakara, and the research student is Mr. M.L.R. Meragalge. The next project title is Characterizing Landfill Gas Emissions, Experimental and Numerical Investigation. And the principal investigator is Dr. D.T.K.K. Chamindu, and the co-principal investigator is Dr. M.C.M. Nazvi. I'm afraid that team is also not here today. The next project, process development and assessment evaluation of microalgae-based biofuels and value-added products. And the principal investigator is Dr. T.U. Aryadasa, and the co-principal investigators are Professor R.A. Athalage and Engineer P.H.V. Nimarshana. And the research students are Mr. V. Celian Arachi and Mr. R.G.M.M. Premaratna. And the next project is protection of heritage monuments and landmarks of national and international importance in India and Sri Lanka due to direct lightning strikes, traditional and scientific methods. And the principal investigator is Professor M. Fernando, and the co-principal investigator is Dr. S. Kumara. And the final project obtaining the certificates would be probing the provenance and distribution of heavy mineral placer deposits in coastal areas of southern India and southwest Sri Lanka, a new multi-mineral proxy approach to understand source-to-sink link. And the principal investigator is Professor S.P.K. Malaviarachi, and the co-principal investigator is Dr. P.L. Dharmapriya. And the research students are Ms. A.M.G.K. Vijayaratna and Mr. U.G.A.M.P. Abhevardhana. Professor Malavi Arachi will also be joining online, so the team is not here today. Thank you, sir. To mark the end of the inauguration ceremony, let me invite Mrs. Geethani Kannangara, Director, International Relations, Ministry of Education, to propose the vote of thanks.
Honorable Chief Guests, other distinguished guests, ladies and gentlemen. I deem it a great privilege and an honor to be the one who extended the vote of thanks on this memorable occasion. I extend a hearty vote of thanks to our Secretary, sir, for spending your valuable time here with us to grace this occasion. Sir, we had opportunity to hear your thoughts, and I'm sure this will encourage us in all our future endeavors. From the depth of our heart, we thank Mr. Vivek Gupta, First Secretary Education, Indian High Commission, for spending your precious time here with us. Sir, indeed, you add glamour to this event. I mention my deep sense of gratitude to all the researchers who have successfully completed their research, including Indian counterparts, and special thanks goes to who have come in person here to present their research findings. I also want to thank our moderators who accepted our invitation to enlighten us through imparting their knowledge. On behalf of the Research and Innovation Division, my sincere gratitude goes out to all staff officers of our ministry, chairmen and CEOs of the CEOs and heads of our institutions who have blessed us with their presence today. Next, I would like to thank the torchbearer of this event, our Additional Secretary, Nadika Madam, for leading and inspiring us towards achieving the goal of conducting this event. Above and beyond, let's not forget the backbone of this event, National Science Foundation, who went an extra mile and made the dream a reality. And a very big thank to Dr. Mahesha for your zealous support. And needless to mention that SLEDA and the sound providers who take care of providing all the facilities so that we can proceed this event successfully. An event like this cannot happen overnight. The wheels started rolling weeks ago, and I take this opportunity to thank all my staff who have given their fullest support to make this success. and special thanks to Sashi. Thank you again to all assembled here for making this event a grand success. My sincere apologies if I have missed out on somebody. Thank you.
Ladies and gentlemen, that marks the end of the inauguration ceremony. The organizers have arranged to take a group photograph of the research teams. Therefore, all members of the research teams and Secretary Mr. M.N. Ranasinghe, First Secretary Mr. Vivek Gupta, Additional Secretary Mrs. Nadika Vataliyadda, and the Director General of NSF, Dr. Sephalika Sudasinghe, are kindly requested to proceed to the front lawn for the photograph. All the other guests are invited for refreshments at the cafe on the ground floor. You are kindly requested to be back here by 10 o'clock. 10 o'clock for the technical sessions. Thank you so much. I have to save it. Here, here. Hari madam. That is, this was the one that granted us. Yeah. You can just say now kids and everything. Yes. Welcome back, ladies and gentlemen. We are about to start the technical session. The technical session will be conducted under three categories. Category 1, plant-based medicine. Category 2, metrology, space research, industrial electronics, robotics, and automation. And category 3, food technology, energy, waste, electrogeometric, and oceanography. And as panel members for the technical session, we have three eminent scientists, Professor Ajit Dialvis from the University of Moratuwa, Dr. A.M. Mubarak from the Sri Lanka Institute of Nanotechnology, and Professor D.A. Santrigoda from the University of Sri Jayawardenepura. Professor Santrigoda will be joining us in a short while. And representing the National Science Foundation, Dr. J.G. Shantasiri from the International Affairs Division and Engineer Mahesh Disanayaka from the Research Division of NSF. I am pleased to invite Professor Ajit Dialvis to give an introduction and continue the technical sessions. Over to you, sir.
Hi, Bowen. Thank you, Dr. Inoka. So, I suppose we come to the session which is quite important and relevant. That is, you started this journey in 2017, and here we are today, 2023, 17 projects, and it was interesting to listen to 28 journal papers, 201 conference publications, and 21 abstracts. But that's a little bit of an interest also, considering the current economic situation and so on. How can this translate these research findings, research to economic gain? How do we do that? And so the potential, that is quite important. And also we will be looking at, and all of you, when you're presenting, it's also important to just think about a bit. We are talking around 12:50. At the end of all these presentations, we will be having a way forward discussion, perhaps what lessons to be taken in and directions to be taken, because this is not to be the end in a sense of research, right? And also the partnerships and the collaborations formed, this should not be an end. Perhaps sometimes this may be a beginning as well of a new journey. So with all that in mind, so think about something that you can put on the table for consideration for the way forward as well for the India-Sri Lanka research collaborations. So how we will do this three categories at this technical session is while we manage, we have 10 minutes given for each presentation for each research project. And that will go as per the schedule. And considering, of course, the management of time, we will be having at 8 minutes, single bell, and then double ringing at the 10 minutes. So let's hope we will be able to manage time. Know a little bit ahead, which is of course a rarity, but it's interesting. We have a little bit extra time. The point is, let's share the work done and the most interesting lessons learned, and also inputs to the way forward. So that's the objective for us as panel. So Dr. Professor Tantrikota will join in short, but I have Dr. Mbarak with then two from the NSF, Dr. Sri, and as well as Engineer Mahesh. Right. We thought there's a small assessment also that will be done. That is, and it'll be given later on for each project for some self-evaluation as well. The idea being all these projects to be looked at from the commercialization angle, or what important information that has come out may help in future. So we are looking at two aspects, that is the knowledge generation from the project, as well as the impact of the project. So while we also may do this evaluation, you will be also asked to do a self-evaluation of your project. And perhaps those colleagues who are joining online from India, as well as the Sri Lankans, also the researchers who are joining online. So we will share this for that self-evaluation, but we will also be looking at it from that angle, looking at how to formulate research projects and how to fund research projects and to generate benefits to the economy at the end of the day, right? The inputs given, how we can get the investment reaped through like return on investment. So that's a learning lesson as well while we all listen to your presentation. So with that, I suppose we can start category one, the first one, the plant-based medicine. The first presentation is high throughput genotyping to expedite the genetic characterization and dissection of important agronomic traits of tea. Professor R. Nathanayake from University of Kelaniya.
Good morning to you all and happy Friday. So let's move on. The topic, I'm not going to say the whole topic. What we were doing was about genetics, combining breeding, and genotyping and phenotyping all together on teas. Okay, so I think I'm audible enough, right? So we started the project in 2018. It was a 3-year project with the pandemic and all. So it went for 3 years. Originally, it was planned for 2 years. And end date was 2022. Budget utilized was nearly 48 lakhs. And this is our fantastic research team. So Dr. Ram Kumar Sharma from India being the PI, and Upendra and Vishal and all these people, and from TRI. So this is like a 3-way collaboration, University of Kelaniya, myself, and TRI representing Dr. Mahasin Ranatunga and Dr. Dhananjay, now he's a doctor, as a result of this program actually. Dr. Amarakon and Dr. Gunasekara from Sri Lanka. The general aim, actually our overall aim was to reduce the selection time of tea. What is meant by selection time was, we know when you plant a tea, you have to wait until it grow up and do the biochemical and all the other parameters, yield parameters and all. So it takes nearly like a considerable amount of time. What if we have a DNA piece that can tell us that this particular DNA fragment is responsible for this particular character? Then we can reduce the time of the evaluation and the cost associated with the field evaluation. You know, when you plant things in the field, you have labor cost and so many things associated. So our big aim was to find the DNA markers for particular characters. So that when the plant is in 2-leaf stage, all we need is just 1 leaf to extract DNA and move on. Okay. So these are the general objectives. I'm not going to go through everything. Basically, we in Sri Lanka, the germplasm accession had like 400 accessions. We don't know really by looking at whether they are the same or not, because tea looks the same, right? So are they really genetically distinct or not? We don't know unless you look at the DNA, right? So that was our major aim, looking at the whole population level and look at the hidden messages in DNA. Basically, what we are doing is from phenotyping, looking at the phenotypic characters, our morphological characters, to the genotyping, genetic characters, which will help us in marker-assisted selection. Okay? So these are some of the tea accessions that they maintain really nicely. It's a commendable job they are doing in maintaining these lines in Sri Lanka and both in India as well. So our party, we had this mapping population of this. You can see the red tip leaf tea and the normal tea. There's a hybrid population that is generated from these two, and population of nearly 200 individuals are there. And all were characterized in terms of biochemical, morphological, yield, everything. Kind of a major, most of the phenotypic characterization has been done by traditional classical methods. So now then, from phenotypic to the genotypic genetics, that is where I was involved. Actually, that is what my interest was. So this is kind of an example where you see these patterns. When you see these particular DNA patterns, you can predict if it's statistically significant. If it happens always, you can tell just by DNA piece of the single leaf stage, you can tell the plant is going to be resistant to blister blight or whatever, right? So this is an interesting graph that we data that we found. I'm just going to show only a little actually. So here what we are seeing is the anthocyanin production. So you can see the parental lines, TRI 2043, one variety, another variety over here. And this is the whole population, how they behave in terms of anthocyanin content. The beauty is, you can see one parent having high anthocyanin content, and we have the offsprings, Blamai, Patau, right? So we have offsprings more than the parental lines, which is called hybrid vigor, which is very interesting phenomena in genetics. So what it tells is the highest anthocyanin content tea we have now, we have better ones, right? So to move forward, idea is why who cares about this anthocyanin? Because this is a specialty tea where Kenya is the leader right now. They are producing something called purple color tea. Our target is to move forward that direction. Another interesting thing we found is variation in caffeine level. That is also interesting. You can see two parental lines in different colors. There are low caffeinated varieties as well as high caffeinated varieties right now. Why low caffeine is interested? Because it's a big industry right now, decaffeination. Because people don't like, some people don't like caffeine. So why some people like caffeine? So we have these varieties as well. And it's been characterized. Yield parameters were measured and determined, and you can see the variation again. Unfortunately, some of these high anthocyanin producing ones are not really high yielding. So that is the direction that we have to work right now. Then move into the marker work. So basically you extract DNA, and this is where our Indian partners helped us, because we do not have the technology to go for high throughput assays in Sri Lanka. Thanks to Dr. Sharma and our student, Dr. Dhananjaya, they worked really hard on it. And they have found that the markers, what is called transcription factor family associated marker, I know to some it may be like Latin. The transcription means you have DNA, you get protein, transcription and translation, that is the process. So if the gene is transcribed, we know that is a real gene, that's a useful trait. So those characters associated markers were identified. You can see abiotic stress related genes, biotic stress related genes, and secondary metabolite related genes, and leaf development related genes have been identified. Now we have the markers to tell that these particular fragments are really helpful in identifying these particular traits. Okay. At the same time, that is like little segments from the genome, whole genome, like whatever the number, 12 or whatever the chromosome numbers we have in the little fragment from the genome. It doesn't tell us a good picture about the whole genome. So what we did was we went for the whole genome sequencing assay. Again, India has the Dr. Sharma's lab, IHPT have this machine, NovaSeq. I think whole India, if I'm correct, it's like very few machines they have even in India. So they use this fancy technology. We used 95 selected accessions, scanned the whole genome and identified the related markers, SNP genotyping. So this is one result that using SSR and all these data. What you can see here, it's a, what is it called? It's gone. Okay, right. What we are seeing here is a bar chart, is a Bayesian clustering posterior probability analysis. Don't worry about it. I will take you through here. This axis shows the how these individuals belong to each category, but the bigger picture is you can see group A mostly red, that is Sri Lankan sample, B and C mostly from India and other cultivars. So this is what we see. Something very problematic is you can see most of the Sri Lankan cultivars are genetically not so diverse, which is not a good thing, because if a sudden disaster comes, if a sudden disease comes, all the accessions might go up, right? So this tells us we need to improve, we need to keep hybridization, getting the more genetic background to the tea. So this is one of the interesting findings, and these are the economically aspect, socio-economic impact. We can go for purple tea, yellow tea, high caffeinated, decaffeinated, whatever the tea we like, and the outcomes, students and papers, and especially most of the important thing is identification of the varieties and the markers. So these are abstract, I'm not going to talk about it. So that's it. And so the MOU is still effective, so the collaboration is still going on, and we wish if we can go for the second term actually. Anyway, thank you so much.
Thank you. We'll move on to the next paper. That is renewable chemicals from the plants of Indian subcontinent. Isolation, self-assembly and utilization in drug delivery, medicine and catalysis. Dr. M.T. Napagoda, University of Ruhuna.
Good morning. It's a great pleasure to share some of the key findings of our collaborative research project titled Renewable Chemicals from the Plants of Indian Subcontinent, Isolation, Self-Assembly and Utilizations in Drug Delivery, Medicine and Catalysis. So the investigators for this joint project are from the University of Ruhuna, including myself and Mrs. Kanchana Vijaysekara from the Faculty of Allied Health Sciences. And from the Indian side, it was Professor Gopal Bag from the Vidyasagar University, West Bengal, India, and Professor Jayanta Kumar Ray from the IIT Kanpur, India. And the duration of the project was for 3 years, but due to the pandemic situation, it was extended till June 2021. And if I give you a brief introduction to the topic, so this, as some of you may have heard, that terpenoids are important plant secondary metabolites, and these terpenoids, they can be used for different purposes in different areas in science and technology, because they have a vast array of biological activities. And one of the important feature about this terpenoid is that they have a natural tendency to spontaneously self-assemble in liquids that would yield different morphologies, such as vesicles, tubes, flowers, petals, and fibers of nano to micrometer dimensions, and also supramolecule gels. And this potential can be used for various applications. So the objective of this project was to evaluate the biological activities of some commonly available plant species in India and Sri Lanka, and to isolate bioactive secondary metabolites, particularly terpenoids, for the assessment of their self-assembly properties in different liquids. And under that, there are several specific objectives, okay, which includes isolation of different plant metabolites from the plants that are available in Indian subcontinent, especially from India and Sri Lanka, and the purification by various chromatographic techniques and characterization by various spectrometric techniques, and then to study the self-assembly properties of these isolated compounds, and study the morphology using different techniques, and then to identify the biological activities and focus on how to utilize these self-assemblies for various purposes like drug entrapment, then generation of hybrid materials, or recyclable heterogeneous catalysis. So under that, first we have collected plant materials from India and Sri Lanka, and then these extracts were initially tested for biological activities. Especially we have focused on antimicrobial and xanthine oxidase inhibitory activities, and then the activity guided fractionation was carried out to isolate bioactive metabolites. Especially we have focused on isolation of terpenoids, and then the structure elucidation was performed, and self-assembly properties of these compounds were conducted in different liquids. And then the morphology of these self-assemblies were studied by different techniques. And if I summarize some of the major findings, okay, we were able to find out isolate argininic acid from heavy wood of Terminalia arjuna, and the self-assembly properties of argininic acid were studied in DMSO water binary liquids mixtures. And the minimum concentration of argininic acid necessary to form vesicles, this is called as critical vesicular concentration. It was also determined and reported for the first time. And this critical vesicular concentration of argininic acid in DMSO water medium with different ratios of DMSO to water was successfully determined using pyrene as an effective fluorescence probe. And here you can see these self-assemblies. Okay, and similarly, we were able to determine the critical vesicular concentration of monohydroxy triterpenoid called as olenoic acid, and it was determined using DMSO water at various ratios. And a decrease in critical vesicular concentration values were observed with increase in the percentage of water in the binary liquid mixture in both of these terpenoids we have studied. And this is another example. This is the preparation of sodium and potassium salts of nanosite triterpenoid maslinic acid. Okay, so these potassium and sodium salts were prepared from maslinic acid, and their self-assembly properties was studied using in aqueous and aqueous organic binary liquid mixtures. And here you can see the high resolution transmission electron micrographic images of these dried self-assemblies of sodium mesylate, this one, and potassium mesylate in different solvent. And these vesicular self-assemblies can be utilized for drug delivery applications because they have the potential to carry the pharmaceutical compound to the targeted site safely, and also they can able to retain its desired therapeutic effect. And our study demonstrated that sodium and potassium mesylate self-assemblies, they can entrap the anti-cancer drug doxorubicin, and the subsequent release of this drug in water can be achieved by changing the pH of the medium without using any disrupting agent. And this diagram shows the fluorescence emission spectra of free and entrapped doxorubicin inside the vesicular structures from sodium mesylate and potassium mesylate. And another part of our study was to study the biological activities of these terpenoid compounds. And we have specifically focused on antibacterial activity studies. And here we have mainly used five terpenoids: stigmasterol, bixene, lupiol, androgopholite, and muscarinic acid. And interestingly, we were able to detect a diverse antibacterial activities with these compounds. So, especially a diverse inhibition pattern was observed with stigmasterol, bixene, androgopholite, and muscarinic acid against E. coli. So, this inhibition was highly significant because the inhibition occurred in lower concentrations, but at higher concentrations were unable to inhibit the bacterial growth. And these observations, they resembled a phenomenon called eagle effect, which has been referred as a paradoxically reduced antibacterial effect of some antimicrobial agents in high doses. And to the best of our knowledge, this is the first report of eagle effect in any naturally occurring phytochemical. So here you can see the results of our experiments. Okay, so we have performed MIC evaluation, and also we have determined the MBC. So these experiments confirm the eagle effect. Okay, so based on the findings of our project, okay, we can draw a few conclusions. Okay, so this investigation revealed that medicinal plants available in Indian subcontinent are rich in phytochemicals, which can be used in various fields in science, particularly as antimicrobials. And this study has revealed eagle effect of antibacterial activity in several terpenoid compounds isolated from plants in Indian subcontinent. And this is the first report of such a property in plant secondary metabolites. And this study enabled to validate different methods used to determine the critical vesicular concentration of terpenoid molecules and hence their self-assemblies. So the vesicular self-assemblies of sodium and potassium salts of muscarinic acid were capable of encapsulating the anti-cancer drug doxorubicin in aqueous medium, and this property could be harnessed for targeted drug delivery applications. And also this eagle effect observed for some phytochemicals contributes to new knowledge, and actually we need further experiments to study the mechanism in details. And we have one publication, it is still in review in the journal Biomaterials, and we have several other publications, and we have presented several abstracts. Okay, and thank you very much for your attention.
Okay, next presentation will be made by Professor P.A. Paranagama from Calgary University. The topic is bioprospecting Indo-Lycanic fungi from mangroves in Nigambu Lagoon in Sri Lanka and Gulf of Habat, Gulf of Kutch from Gujarat, India, and untapped treasure trove for discovery of special structures and bioactive compounds. Professor Paranagama.
I'm sorry, I changed my name for a while. So Professor Paranagama has a family emergency. Yesterday night she called me. Can I present? I just made slides yesterday night. I may have mistakes. I'm sorry. Okay, we'll see. So the this project, I was a co-PI. Where I involved in DNA type work. I has nothing to do with chemistry. So the project started in 2018, January again. And principal investigator was Professor Priyani Paranagama from our university. And Ramani is our student, and Nipuni is here. She's also our student. Principal investigators from India are this Kiran Khalia and Abhijit. They visited us. And allocation project, we spent nearly 85 lakhs. This is the expenditure summary, financial summary. And the objectives were, this is like the, we have lichens, that is a fungal algal association. What we are looking at is fungi living in addition to these two organisms, the fungi and algae. So some other fungi, what we call Indo-Lycanic fungi. So Professor Paranagama's group is the first to study such thing in Sri Lanka actually, and this is an extension of this continuous life of her. So they are what we were interested in, isolating and identifying this mangrove ecosystem associated Indo-Lycanic fungi. Why mangrove is because mangroves are a harsh condition. It's a very hard condition, and lichens is another harsh. It's a very limited resources living, and it's a very limited resource for any living creature. Fungi living in a mangrove associated lichens will be a really interesting part. That was our hypothesis, and that is what we were looking at. So the basically extract DNA and identify the fungal species, and then go for the secondary metabolite as what Mayuri has been explaining, I guess. So the so the main project at glance is looking at the secondary metabolite, looking for bioactive properties, bioassay guided fractionations, and identify novel compounds, and looking for in vitro and in silico assays were also done. That is something very interesting part. Another sub-project, while we were studying on that, I had another grant on polythene degradation. So we were discussing why not trying that one as on this as well. So we got the permission and tested the polyethylene degradation ability of Indo-Lycanic fungi as well. So these are some of the pretty lichens we collected, and all the lichens were identified with the help of Dr. Gautami Veerakorn, eminent lichenologist now in UK. So these are the lichens, and these are some fungi that we isolated and identified. And I know this figure is miserable. What I'm showing is the diversity, high amount of species diversity we found even in the lichens actually. And then LCMS dereplication metabolite profiling was done, something called that. I don't know what it is, but what it's supposed to be is identifying the chemical profiles and all. So these are the some of the interesting activities that we found: bioactive properties, antioxidant properties, and the anti-inflammatory activities, even less than the control aspirin, and IC50 value, okay, and the tyrosinase inhibition activity. These are some of the very interesting findings. In addition to that, we tested the antibiotic properties of some of the Indo-Lycanic fungi. Only little data is here for the clarity actually. There were a lot. So the you can see here, it's very clearly some of the fungal extracts are showing an inhibition shown for all four bacterial types, E. coli, Bacillus subtilis, and all these, right? So these are the now chemical compounds that we isolated and identified and characterized structure with their structures. And the sub-project that is conducted was trying to find a solution for polythene accumulation on Earth. So we tried looking at whether these particular organisms have the ability to degrade polythene. Why? Because they are if these fungi can living in such a harsh condition, why not on polythene? Is it possible? Because there are some evidences with the previous literature. Tested series of studies. So I will show you only the how did we confirm the polythene degradation by these assays. These are some of the SEM images that the control, you can see it's nice and smooth. When the fungus is treated, you can see the cracks and holes are found in the surfaces. FTIR peaks and the functional group changes compared to the control. This says that the polythene surface then has been changed. Okay? And then the student conducted some real field assays as well, collecting from water samples from this Karadiara, whatever that garbage dump, and then they did the assays in the field as well. She conducted this field trial, like utilizing that water and inoculating our fungi to see whether it as a consortium can have an effect. So the outcome is we have a collection of lichens and endogenic fungi maintaining in the department right now. And Perenniporia was identified as outstanding agent for polythene case. And in addition to that, so many fungal strains and chemicals properties have been found, and socio-economic impact. Here these are untrapped. We have been telling this ever. I'm really sick of this word. We are a biodiversity hotspot. We say hotspot every day. Utilization of this hotspot is really below the average actually. So again, I have to say that this is a untrapped resources are plenty, and this is one such occasion only. Saying that these have this much of metabolite profiles, and next step what we should do is try to get the assays done in Sri Lanka, and with, of course, with the collaboration of India and all. We should start moving, having our own systems as well. So these are the publications we are not very much interested, and the future work is just this is something I would like to propose. Like something I learned from India from my my collaboration with Dr. Sharma. Actually, they have this something called Pentagon system, amazing system. The whole thing is connected in one place. Genetic diversity and genetic resource study in one place. The same single institution, IHPT. Genetic resource diversity study is done in one group. That is my Dr. Sharma, and in this case, this people is another case, I guess. So then natural product chemistry and assays, natural product section, you find the chemicals, you find the diversity, you try to preserve them in the Himalaya region in that case actually. So this is the system throughout the several organizations that I have found actually. Natural product chemistry and assays are done, whatever recommended from the genomic people, you do the assays here, and then best chemicals or best products will be going for genetic testing and animal testing unit, animal testing, genetic testing, in vitro, in vivo, cell culture, everything. And then if there is something, go for the commercialization unit. So this is something I truly like. One unit in Sri Lanka, that will be more than enough. We should not allow these things to happen every part, district in this little island, just one unit for this country. That's it. Thank you. And thank you.
Thank you. Next, in vitro and in vivo epigenetic investigations on anti-cancer and radio-priming effects of different parts of Mangifera indica, India, and Mangifera selenica, Sri Lanka, and impact of green silver nanocomposites by Professor K.H. Tennakon, University of Colombo.
How do I get the
Make another.
can't tell you. Thank you to the organizers, the ministry people, as well as National Science Foundation and panel and all the researchers who would be listening. I have a very, very brief presentation because I went along the format which was given. So the title is there, you can read it, basically looking at Mangifera selenica, which is our endemic mango, and Mangifera indica, which is the common mango, and we were trying to look at certain anti-cancer effects of these. And also there was a part which was where Indian team was going to be responsible for developing green silver nano composites, which did not happen in the life cycle of the project, but that is now happening because there were a lot of problems they had during the COVID time. They were more affected than us. So basically what happened was that we had been working on cancer biomarkers, as well as anti-cancer drug discovery for many years. And this Indian team came across some of our publications on Mangifera salinica, where we had demonstrated that we had isolated active compounds, which are active against certain forms of breast cancer and ovarian cancer. So they wanted to collaborate with us, and we readily agreed under this program. So the duration was 3 years, and it was extended for further 9 months due to the impact of COVID. And there were some delays in settling grant finances and so on, because the experiment work continued beyond that date. So actually the date of completion was 31st of December 2021. And we had a total allocation of about 3.3 million, and some 128 rupees were left behind, 180 rupees, which we had to reimburse. So that's all which we saved for the ministry from that project. And when I asked, is this small amount has to be reimbursed, I was told, yes, it has to be. So I think even the check may not have been worth on which it was written would not have been worth. But total that we had initially requested was 5.6 million. So we were like 2 million short to carry out all the work, but we had to recast work, especially in view of the COVID pandemic. So the research team was from the IBMBB in Sri Lanka, myself, Professor Samira Samarakon, and he was the one who was live wire behind this. Actually, I lent my name, more or less my name, to get the grant, as well as to report the finances and so on. So he was the one who was on day-to-day supervision. And Dr. Mirani Trivira, who was a research scientist at that time, and now he is an academic in the medical faculty, and Nirmal, who is here, who is the PhD student. The group who contacted us were from a private university in India, which is called Sastra University, School of Chemical and Biotechnology in Tamil Nadu. And Dr. Devipriya Nagarajan was the PI, and then there was a senior person, Dr. Valivel, and also their PhD student. They visited us, and Samir and Miran visited India. So the general aim was a comparative evaluation of anti-cancer and radiopriming properties of medicinal plants available in India, that was Mangifera indica and Sri Lanka Mangifera salinica, in order to improve the treatment modality of lung cancer patients. Now, you know that cancer is a deadly disease, and many people are affected with cancer. The prevalence of cancer keeps on increasing. Even with a lot of advancement in therapeutics, so far the prognosis has not been that good, and lung cancer is one of the deadliest forms, and it is a second commonest cause of cancer deaths. So there are very few treatment modalities which are effective for lung cancer. If you get a diagnosis of breast cancer today, you could say, okay, it's like common cold and everybody gets it. 1 in 8 women in the US or whatever. And then if it's early stage, it's very easy to treat and you just live a normal life. But if you get lung cancer, your days are very, very much numbered. So that's why these our Indian investigators were very much interested in lung cancer. Since we had not worked on lung cancer with Mangifera, we also agreed and carried out the project. So when we look at the specific objectives, I have put the specific objectives of the Sri Lankan team. And basically it was to collect, identify and authenticate the plant, and then prepare solvent extracts, do phytochemical analysis and cytotoxicity on lung cancer cells. And when we do the lung cancer cells, we also at the same time compare with normal lung cells. Otherwise, just because something can kill the cancer cells, we have to make sure that it will not kill normal cells. So that is one of the shortcomings that I find very often in various research proposals and so on, that they will not use normal cells to show that they are not cytotoxic to normal cells. Then we were planning to exchange solvent extracts between the two teams, and I also isolated phytochemicals, and then evaluate anti-cancer effects on cell lines and look at epigenetic modifications. Epigenetic is something where changes occur to the gene. Now we have the nucleotide sequence, our genome, the nucleotide sequence, if we take, if there's a change in that, in a nucleotide, we say there's a mutation and so on. So that can affect your gene expression. Epigenetic is where without changing the nucleotides, whereby causing changes in gene expression due to certain changes like histone modifications, methylation, and so on, the gene will not produce the protein. It will not produce the RNA, and it will not produce the protein. So by doing DNA sequencing, you cannot identify pure DNA sequencing, you cannot find epigenetic modifications. And now the tendency now is to look at in the diseases as well as in this kind of work, to look at epigenetic changes, because it seems that our nucleotide sequence has not changed. It is like the nucleotide sequence is, our genome is like a book. A book is useless if you just keep it on the table, you have to read it. So the way it is read has been changed. So if it is being read wrongly, then you get wrong results. So this is like that. Epigenetic changes would be like that. Now, then we had to also revise certain objectives, like especially those in relation to nanocomposites, because we were not receiving nanocomposites from the Indian team, and we added the structural elucidation of the isolated compounds on our end, and also to evaluate the mechanism of anti-cancer effect of the isolated compound. So this is the project at a glance as per modified work plan. I explained that because in the title it says nanocomposites and radiopriming, and you will find that it has not happened. So basically both teams were doing the same thing. We were working on Atamber, the endemic mango, and they were working on a variety of mango in India. Collection, identification, and authentication of plant parts, then prepares different solvent extracts, and then phytochemical-based bioacid-guided fractionation, and so on. And then, of course, we looked at the cell line models. That was our part. And the Indian team was going to look at the in vivo part. They were going to work on animals. And so we added this NMR spectroscopy and isolation of compounds. And our project output is that we had academic visits, 2 PhDs, one here and one in India. They are being completed now, and joint publications. And we identified anti-cancer activity against lung cancer in endemic mango and common mango. And then we evaluated the mechanisms of anti-cancer activity. Sorry. The 2 joint publications are listed there, and then other publications by ourselves and by the Indian team are on the way. And this is one of our publications, which is a review article. And then this is the other one, which is the research article, again on epigenetic modifications. They have joint authorship. So our way forward is for these PhDs to be completed. And if the funds are available, you need to do the safety and toxicity studies, then preparation of anti-cancer formulations for preclinical and clinical trials, and also elucidate the mechanisms of nanoformulations, which are being prepared by the Indian team. So that will require further funding and for other people to take on and do, because I have now retired, so I have handed over the mantle to the Dr. Samir and the others. So actually I had to refresh my brain to come and do this presentation here. So I'm not in this program anymore. Thank you.
We'll move on to the next paper, electronic and photovoltaic properties of two-dimensional hybrid organic and inorganic perovskites, presented by Dr. MLC Artigala, University of Sri Javadam.
Good morning, everyone. I would like to thank NSF and also the Ministry of Science. Now they have added lots of words to that ministry. To all of you to have this symposium and giving us a chance to talk about our findings and also way forward. Okay. I think Professor Jha is also here online via Zoom. Good morning, sir. Sri Lankan team represent myself as the principal investigator. I'm Lilani Artigala from Department of Physics, University of Sri Jawadhanapura. And under this project, 2 MPhil students formed, not actually, now they have graduated. Nipuna Lochana Aditi, he is here with us. He completed his MPhil degree in 2022. And Dinuka Ratnasinghe, at present, he is at NBRO as a senior scientist. He graduated in 2021 with an MPhil degree. And Sampath, he was from University of Moratuwa. He was with us for like 5 months, but he joined the first trip to India to begin the project. And from the Indian team, we have Professor Jha and his PhD student, now Dr. Somnarayan. So here I have put a picture. This is Dinuka, Nipuna, me, and Professor Jha and Som. So if I mention you briefly what is this project is about, we wanted to develop more stable perovskites. This is, a material that we can use in solar cells. So we wanted to actually find out such material as rapid rise in energy conversion, efficiency, low degradation, and higher moisture stability to build thin film solar cells. Now there are several versions. Here I have mentioned multi-junction, low-cost thin film solar cell structures. So basically we have developed materials, and also we tried considering some modeling softwares to form thin film solar cell structures with high efficiency. If I show you the brief, summary of the project work. So Indian team actually contributed in the theoretical studies side. So we have done the modeling and the simulation. So this is open. We have submitted another proposal to continue the experimental work. So I will discuss what we have reached. So instead of doing it in experimental lab, we have done it in a computational way. So developing solar cells using the materials that they have developed. So if I show you all the objectives from both sides, here we can list like 8 objectives. Mainly we wanted to understand these low dimensional, that means 2D perovskite materials. So no one has calculated some of the required properties. So we got the help from the Indian team to figure out some of these parameters we need for the solar cell modeling. It's not an easy task. It took a long time because actually they have developed several materials, and we have to find out a way, to include in which part of the solar cell. Like maybe we have to find out different type of solar cell structures. So then they have to again, remodel the material and find new parameters. Here I wanted to show some results, not everything. So you can see this is like one of the structures we have built numerically, and we done the simulation using SCAPS 1D. So you can see 2-dimensional MAPI, 3-dimensional MAPI, 2D MAPI. So all these junctions, so these are semiconductor materials. So all these junctions we have to study carefully when we are building the solar cell. So we can predict some, efficiency values, short circuit current. There are some factors that we can use to identify whether solar cell is a good one or a bad one. So Not only that, we try to build multi-junction solar cells also using the computational environment. And then we got some good result. So not only the computational work, we have to actually discuss the theories behind it and the transport phenomena, because then the findings that we have can be used later with other research groups. So some more details. This is actually one of the other new material they have developed, delphosite, copper alumina. So recently, actually, we got several citations for these materials and the work we have done. Okay, as the project outputs, I can list these two mainly. So we have added new knowledge, and also I think these results that we done using the computational environment can be done in experimental way also, and confirm whether we can go or use 2D solar cell materials like the one that we developed, perovskites for low degrade and high efficient solar cell. In terms of publications, all these are joint publications for index journals, local journal publications, and some conference proceeding abstracts. So these are some details. So suggested way forward, actually sorts of human resources developed, actually two MPhil degree students, one PhD. So they can actually provide the necessary guidance to go ahead and work with the other research groups. So we are looking forward to have that grant so we can build the solar cell. And some interesting fact, we found these thin film solar cells, built using perovskite materials can be work under low lights. So even that means under this light also we can find some sort of solar cell to work. Okay, that's it. Okay.
Thank you, Dr. Atagala. Thanks.
Hello, sir. Thank you.
Hello. Next presentation will be made by Dr. Gamage of Peradeniya University. Topic is enhancing the formability and automotive materials at elevated temperatures and selection of green lubricants for manufacturing. Dr. Gamage.
Very good morning, all of you. So I'm Lakshita Meragalge actually. So I am presenting the things that we have done on behalf of Dr. Pramila Agamage. So topic of our research work is enhancing the formability of automotive materials at elevated temperatures and selection of green lubricants for manufacturing. So this was 2-year project, and we completed around 3 years. And this is our research team. So from University of Peradeniya, Dr. Gamage and Dr. Nanayakkara and myself as the research assistant here. And from the Indian team, we have Dr. Ganesh Narayan and Dr. P.S. Robi from Indian Institute of Technology, Guwahati. So with this research team, we conducted the research, and this is the aim of our research work. It is to find a sustainable solution to enhance the formability level of automotive sheet materials. So to succeed this project, we have two main objectives. First one is to investigate the effect of sheet material forming parameters such as lubricant, temperature, and tool parameters on the formability of forming aluminum AA2014 series. So this is aluminum alloy series. And the second objective is to reinforce the experimental findings with a simulation model, finite element simulation model. Right. So if I go through the background of the research briefly, so this is mainly deal with the automotive manufacturing sector. So with the growing current challenge, current trends, automotive manufacturing industry is moving to sustainable solutions by reducing the total weight of the automotive vehicle. So they are resizing the vehicle and try to reduce the weight. And the next solution is reduce the weight by replacing existing material with light materials. So by the way, by reducing the weight, they can achieve full economy and they can reduce the emission. So automotive manufacturing industry has recognized that 10% reduction in the total weight will lead for 6 to 8 reduction in the fuel consumption. So this is best way to go for a sustainable solution. So mainly they are focusing on lightweight alloys. So the main problem is with the lightweight alloys, the flow formability comparing to conventional materials. So we have to identify new forming techniques and parameter conditions to increase the formability of those lightweight materials if we are going to use those in the automotive manufacturing. So mainly automotive manufacturing sector is focusing the automotive vehicle body panels because it contributes 20% from the total weight. So here we have developed 3 hypotheses. First one is forming temperature and lubricant significantly affect the formability of lightweight alloys. And the second one is sustainable lubricants can be used for replacing alternative synthetic and mineral lubricants. And the final one is finite element simulation model can be used for predict formability limit of lightweight materials. Okay, so this is our methodology what we have done. So initially with the background study, we have selected a material for this study that is AA2014T6 alloy, aluminum alloy grade. Currently, it is used in aircraft manufacturing. Actually, fuselage of the aircraft, this material is used, and it is well suited for use in automotive vehicle because of its high strength to low weight ratio. So we have designed the research work in 2 phases. In the first phase, we change 2 parameters in 3 levels: temperature and lubricant type in 3 levels. And with the results of the first phase, we introduce 2 green lubricants to replace the existing synthetic and mineral lubricants. Simultaneously with those 2 phases, we develop simulation model that can represent the experimental testing by using the simulation model. Then we can save time and cost. So with those 2 steps, we came to the conclusions and results. So here the measurement of the formability we have to do using a forming limit diagram. Actually, you can see the diagram. It is representing uniaxial region to biaxial region. So if we take an automotive vehicle body panel, it contribute these all regions. So we have to check the formability of the material from these all regions. So we have to develop this forming limit diagram, and we have prepared those samples according to the Nakajima standards, and we have done the testing. So this is the place we use resources from the Indian team. We did all the experiments in the IIT Guwahati, India, and according to this experimental setup, finally we did the strain measurements and we developed a forming limit diagram. So I will show the results and outputs here. You can see the developed forming limit diagrams with 3 different lubricant types and 3 different temperature levels. Then we can easily recognize that with the temperature, forming limit curve has moved upwards. That means in the positive direction in the major axis. That means the formability level of that material has increased with the temperature. So we have done statistical analysis with the results, and it is clearly shows that there's a significant impact from the temperature for the formability of that material, and also there's no interaction between lubricant type and temperature. So with the results of the first phase, we developed the second phase. In the second phase, what we did was we introduced 2 green lubricants. First one is boric powder plus canola oil mixture. So boric powder is the solid lubricant, and we add liquid and it is very easy to apply on that surface. And the second one is soap nut. So it is well popular in India, and it has used as a natural cleaning agent from history, and it has good formability properties. Sorry, good lubrication properties. So we have used that one in our study. So it is available in Sri Lanka also. We call it as gas peneler. So we have used that two lubricants to replace the synthetic and mineral lubricants in elevated temperature foaming. So we have got results here. We could replace mineral and synthetic lubricants with the two green lubricants, and we got the same results that we can get from the mineral and synthetic lubricants. So we can say that we can replace that lubricant with the green lubricants, and this is a good find in this research work. So if I conclude the results, as I mentioned, we can say those two green lubricants we can use from uniaxial deformation region to biaxial deformation region to enhance the formability of the material at elevated temperature. So we have selected 300 Celsius temperature here because that was the maximum formability level that we gained in the phase 1. And also we understood that sheet thickness has a significant effect on the formability. Those are some other results we got from the outputs. And number of MS2 layers also has a significant impact on the formability. Right, simultaneously we developed simulation model. What it was, we developed the simulation model that the same as the experimental setup. So these are the details of the simulation model. I am not going to go through all the things. And here I am showing the agreement with the numerical model and the experimental setup. These are graphs we developed numerically and experimentally, and you can see the agreement of those two graphs in each temperature levels. And we did some numerical statistical studies to check the agreement of those results. And according to this mean absolute deviation value and mean square deviation value, you can see the numeric simulation results and experimental setup results have good agreement each other. So here also you can see the experimental samples where the fracture areas are same in the experimental setup and simulation model. So those two numerical setup and experimental setup have good agreement each other. So if I go to the outputs, if I summarize them, so main key findings are those two green lubricants that we can use to enhance the formability level at elevated temperature for those that AA2014 T6 lightweight alloy. So we can check whether these lubricants can be used for the other materials also. And the next one is the developed simulation model, and it has very good agreement with the experimental results. So we can use it without wasting money and time for the further experiments. Right. These are the publications. We have book chapters and conference papers. And this is suggested way forward. It's better we can implement these findings in real world applications. Okay, so last I want to acknowledge all who supports for the research project work, and thank you all.
Thank you. Next, we have dynamic analysis and shape control of inflatable structures for space applications. We have Dr. H.M. Vaisi Mallikaraj from University of Art.
Good morning, all. I'm here to present our work on dynamic analysis of shape controls of inflatable structures for space applications. So it's basically inflatable or deployable structures what we are talking about. I'll go through that in a little while. But as per the instructions given, let me go through the brief introduction of the project. So it's basically, it was done with Indian Institute of Technology, IIT Roorkee, and total cost was around 4.7 million. And to the amount given to the university, I think we save about only 30,000, but with respect to the sort of bilateral collaboration, meaning the visits, I was able to go there, but they were not able to come basically because of the pandemic situation. And then as for the project team, I am the co-PI and Professor Priyandas has been, I'm the principal investigator and Professor Priyandas has been the co-PI. And I had 3 MSc students and 1 undergraduate student. Out of those 3, Mr. Mirunalan, Ms. Sahangi Dasanayak and Ms. Sujika Nadaraja, all of them did MScs and now they are almost completing their PhDs at the leading institutes in the world. And also Ms. Piyumi worked as an undergraduate student and helped with some of these efforts. And from the Indian side, I had Professor Sanjay Upadhyay as from the IIT Roorkee, and Parth Kamalia is the PhD student from Indian side. So let me go through a quick introduction to the project. Since this is a space application, let me go through what is the space application and what is the target, and then I'll highlight the design challenge, objectives, methodology, and what we did, and finally end up with achievements and maybe a suggested way forward. So since this is a general audience, I will not go into detailed technical assessments, but let me try to make it as simple as possible so that you can understand what I'm talking about. So earlier there was a presentation about solar sails, but I'm here talking about solar sailing, right? So the idea here is when you send a spacecraft outside, like out of space, it needs some energy to propel. Whereas if you're sending it as a fuel energy or any motor mechanisms, everything will be adding to the weight of the launch vehicle. So it is actually it's not economically, and sometimes it's not even possible to send it out of space. Right. So now the idea is this is one application where you have a small spacecraft. If you can see at the center here, there's a spacecraft, and you have a large sail which will propel from the basically reflecting the photons emitted by the sun. So this the day-to-day activity. All of you are familiar with what we call wind sailing. So solar sailing is analogous to the same concept. But the problem here, we are talking about very large sails even for a small spacecraft. So what I'm showing here is a 160 meter by 160 meter sail proposed by NASA sometime back, actually in 2009. And I'm giving you here an idea about the scale effect. An actual launch vehicle will be, you will be having only space somewhere here only, right? And I'm not so sure how many of you are familiar with Washington Monument. Let me come back to Sri Lanka. All of you are familiar with Colombo Lotus Tower, which is 350 meters tall. Like, so we are talking about almost half the Lotus Tower and a square that has to be deployed and which will be packed only in a room like this. So that is the challenge that we have, right? So its capacity of the launch vehicles become the bottleneck for these applications. And as I said, even though you have a launch vehicle, only a limited amount is there for the storage. So the idea is somehow you have to pack everything together and put it inside this launch vehicle. So let me play a video to give you an idea. So we are having a small spacecraft here, and you can see like 4 booms deploying this structure, and then you can achieve a much larger scale. Right? So we are going to use thin films and mechanical packaging efficiency, deployment, repeatability, and reliability. And you can increase the storage or like basically compact into configuration. And I think all of you are familiar with what we call origami, which all kids used to play with. So these folding techniques are basically go with that. So if I put you at a very simple problem, like now I have a A4 sheet. If I don't have this much space, what I will do is I'll fold this, and now I can pack it into half a basically A5 sheet. Right, but for us, it's very simple. You can do that, but I'm expecting this to be, when it completely deploys, this should be a flat sheet. Whereas here you can see now it's not coming back to the flat shape. There is due to this plasticity, there's a formation, right? On Earth, you might ask, why do you worry about this a simple piece of paper? But the problem is, as you can see here, if you are to deploy this, you have to have 2 forces applied on this side. And the amount of force you applied governs the entire mechanism, and also the amount of fuel needed for deployment, right? And also the shape as well. So this for solar sailing, the shape is not that important, but if you talk about the reflectors, then the shape accuracy is very important to focus on the light rays. So let me show you a video of where they are actually doing this packaging. As you can see, it involves a lot of creases, right? And packed into configuration, and then it will somehow deploy. This is the concept, right? So the two things I would like you to focus on, one is there are a lot of large number of creases, and then there you can see these shapes here. And in addition to that, these experiments are done on Earth. Then you have the air drag, reduced gravity, and also friction when you do it on Earth. Whereas when you're in space, all those things are different. But unfortunately, unlike many of the other research, you can send something into space, do a trial, and come back. Right. So because of that, people are using numerical simulations. Of course, numerical simulation, you can't just rely on that. You have to always verify. So that has been the overall objective of this project. And then specifically, we were working on this membrane structure. So what you're seeing here is a 3 Kapton specimens, which is actually the space qualified grade material. And then you can see in the middle part where that has been folded and it has a certain angle, right? And then you can do some test. So we are basically trying to investigate how you can do that, how you can model that, and also verify and see whether we can include this in the overall simulations, right? So from University of Morocco side, we were mainly focused on these creases where you can give this shape and the bending of this membrane, as well as the plastic deformation of the hinge or the crease. We were looking into that. And from the IIT Roorkee side, they were looking at this, what we call the membrane wrinkling. It's similar to in your clothes, you get wrinkles. So if you have wrinkles, then there's a shape accuracy effect, right? So the Roorkee side, they were looking at that part. So in a nutshell, at Morocco, what we did is we had a folded structure and a deployed structure, and we wanted to simulate this. And when you are simulating, you have to get this shape, as I showed with the piece of paper. And then we were trying to come up with some sort of a theoretical, basically a mechanical spring that can be simulated instead of modeling all these with then finite elements, which lead the computational time to be very large, right? So we were trying to come up with a rotational spring to replace this so that you can do the simulations much faster. Of course, at different stages, we had to do experiments, theoretical analysis, numerical, and then also verify. So in a nutshell, this is what we did. If I focus only on the piece, as I said with the paper, you get these shapes. And then if you are using a rotational spring, you get much closer results. Right, so I'll not go through the detailed methodology. So we were doing some experiments, and then we did some experimental results with digital image correlation, again with the in-house built technique, because the actual ones or the commercial ones are much more expensive and we can't afford. And then this is an experiment done at California Institute of Technology. NASA Jet Propulsion Laboratory belongs to them. And then we were trying to use that results and simulate and see how our performance works. So what you're seeing in this video, on the left-hand side, you get the one without this rotational stiffness. And on the right-hand side, we have included the rotational stiffness. And in a little while, you'll see there are a lot of colors on these edges, which indicates there's a additional force is required, a stress concentration that means leading to an additional amount of load that you require. So basically, we developed that numerical technique, and we did verify at a single hinge level. And we did a the literature we used to compare. But of course, there are further things to do. So in a nutshell, as an achievement, through the grant, we got a high performance workstation and the software, which is available at the department right now. So we are using that even for the other projects. And we established collaborations with IIT Roorkee as well as University of Colorado Boulder, USA. And then, as I said, there were 4 students, and one of them just graduated from University of Cambridge. Other 2 are studying at California Institute of Technology, and another person at Hong Kong. And we had 4 Q1 journals published, and there are additional conference papers. And we are still continuing on this work. So with IIT Roorkee, we are continuing, and also in the 2019 project, I have something with IIT Hyderabad. So we are focusing on focus. Yeah, with that, I would like to acknowledge these people, and thank you very much.
We'll move on to the next paper, be presented by Professor PBR Disanayake, University of Pennsylvania. Wireless sensor in network distributed detection mechanism for structural health monitoring in civil structures.
Good morning, thank you. So my presentation on the wireless sensor in network distribution detection mechanism for the structural health monitoring. These are the details that I'm not going into the detail. I would like to mention about the Associate Professor K. Patanaike and their group for supporting this research work. We did it at the University of Peradeniya, Department of Civil Engineering. These are the some of the situation that we can see that this is a bridge fail in 2021 in Sri Lanka. This is some of the news article appear in recently. They say that out of the 1,375 bridges in the railway, 850 in the critical condition. Then you may know that the Gujarat bridge was collapsed recently while people are on the bridge. So this is the scene from the failure. The number of deaths was more than 130 plus, right? These are some of the detail. So in history, what we did was, usually when we have a old bridge or the structure, we just simply remove and go for the new one. But we started the research work on this area about almost 20 years before. National Science Foundation provided us the funding for the basic research, and the National Research Council provided us the later for the applied research. Of course, why we want to do in this area, replacement or the new structure is a huge cost for the economy. And of course, presently, not only Sri Lanka, around the world, even the country like US cannot bear this amount of cost, right? It's a national, not a national issue, it is a global issue right now. There we have, after the civilization, we have so many structures have been around, but replacing them are not that easy. That has become a huge topic by now. So a structure like this we have around the world. Of course, we have many in Sri Lanka. So many economy cannot bear to replace this kind of one. What we have to do is structural health monitoring. That is a subject that we are doing. Under this research, we have fine-tuned some of our gaps. So of course, we have to go for the retrofitting, but there are some situations we may have to demolish after the structure retrofitting. So that is the way forward. So just to, as it is a general audience, structural health monitoring is when you have the structure, we have to do a thorough investigation and understand the defects outside, inside, and area. Then if the structure is in weak, we have to strengthen the structure. We call this as the retrofitting of the structure. Just for you to understand, its word come from the health monitoring. Of course, when you have a patient, so that you have to test their blood, urine, and so forth, and prescribe the medicine or the surgery or whatever it is. Exactly the similar way we do as an engineer, but this care, the subject came later, because not like human being. This is came later because now only we have a lot of old structures. Of course, structural health monitoring is a process of implementing the damage detection. This involves the engineering investigation. So by structural health monitoring, of course, understand the health of the structure. We can say that how much we need to go for the spend for the maintenance, and the retrofitting method, how are you going to rectify the structure strength in a way, or some situation we decide we need to demolish it. Then we can calculate the remaining life of the structure, how long the structure can use like, of course, the controlling of load, like a speed limit and so forth. This everything will be decided after the structural health monitoring. By just looking at that, you cannot do it. So if you are not going to do the structural health monitoring, I could not mention even the Gujarat bridge that was collapsed after the repair of 1 week. That means the repairing is like the makeup, right? Makeup won't work for the old people, but it should be a health check, right? So otherwise, this is the happen in India, because this kind of things would have prevented if you have gone for the structural health monitoring. So this is the kind of introduction. So, but our background was where we use a lot of sensors in order to understand the structural health monitoring. So that, of course, we have to connect from the sensor to the data logger, and the data logger closely connected to the computer. So this wire is sometime very long. I remember like when we test the Calne railway bridge, sometime you can remember how long the bridge is. All the way from there to we have to bring the cable. Then we have to limit the number of sensors that we are using. For example, testing like the Calne railway bridge, I mean we have to close the railway line for the maybe 5-6 hours, and the heavy engine is going here and there. It's a different speed, so that how are you going to, we have to capture almost every data within the given period of time. Therefore, it is a big hassle. So that now it has wireless sensors. But when you go to the wireless sensors, we understand that we get the sum of the data, which is we call as outliers. And so this is the area where this is the people in the India they do the computer department. They help us to logarithm and all the testing was done there, but the algorithm developed in India. So you can see that the outliers removed from their software. This is what they help us. So that what we are trying to do is we are going to use the wireless sensor network in future. Now already we have got some. So these are some of publication that I am not going to detail. So actually we have already established the company on this we call as International Center for the Sustainable Built Environment Private Limited. Actually, this is the one of the 5 company that we have developed under the our research work. And recently we have, I'm just putting a little marketing here. Recently we have developed the GAP headquarters. GAP headquarters means those who want to commercialize their mainly the research. We are helping, so that we would like to collaborate with you, but I think this is a good forum. If anybody want to commercialize their research work, we may be able to collaborate and do some of work. For the time being, of course, we have been discussing and working on to develop the several other companies. So these are some of the equipment that we purchased from the grant. And this is, we would like to continue this one. There are a few other gaps that we have identified during this one. I would like to say thank you so much for you.
Thank you, Professor Desanayake. Next presentation will be made on the online mode. It's by Dr. Chaturanga of University of Moratuwa. Topic is design and development of detection and extinguishing systems for forest fire using sensor networks, aerial and ground robots. So do we have hello? Dr. Chaturanga? Yeah. It's muted. We can't hear you. From that end. Hello. Still we can't hear you. Could you please unmute?
Right, sorry. Okay, good morning again. I think I had some problem with getting access to the unmute button. Okay, now it's there. Right, so let me share the screen. Right, I'm Asita Kulasekara. I'm presenting the work by Dr. Damit Chaturanga on behalf of him because he's unavailable at the moment. So let me jump into the presentation for you. Right, so this project has been on the design and development of detection and extinguishing systems for forest fire using sensor networks, aerial and ground robots. And the Sri Lankan team members are mentioned here as given by Dr. Damit Chaturanga. So I'm Asita Kulasekara and I'll be presenting the work for you today. Right. So this work is based on the problem that comes up when we have forest fires in areas that are maybe difficult to reach, especially if you remember in Sri Lankan context, there have been fires in the areas of Riverston and near the University of Sabaragamu as well recently. So problem has been the mountainous terrain and the inaccessibility. So when a forest fire takes place in such an area, the problem primarily is the detection at an early stage. So the primary aspect this project focused on solving is developing this detection and then the subsequent extinguishing solution for such forest fire situations. So in this project, the idea was to develop wireless sensor networks that could detect the presence of the fire in the initial stages and communicate it out to some other listening station. And so this would be communicated to a central node that would dispatch an aerial vehicle, in this case, an unmanned aerial vehicle, what we call a drone, to go and explicitly and accurately detect the location of the fire that has been detected by the wireless sensor node. This would act as a secondary observation and let's say support the initial observation, and based on this finding to send out ground vehicles that can reach the difficult to reach terrain and then extinguish the fire. And this would require secondary support, and in this case the communication and the local navigation would be handled by the aerial vehicle. So this has been the vision in this project. So as the initial parts of this, the proposal is to break this into two areas. The Sri Lankan team would be covering the wireless sensor node and the aerial application, whereas the extinguishing part and the ground structure, the ground robotic structure would be developed by the Indian team. So first part, at the stage one, we had to identify the types of forest fires that are prevalent in the Sri Lankan context and the Indian context. And this was found out based on the international literature and the existing studies that have been done in our two countries. And then the sources of the fire, and primarily the fuels that are present, to identify the type of detection and the type of sensing that is required at the wireless sensor node to better and to rapidly detect the presence of the forest fire. And on this, to establish the smart part, the intelligent system that can grab minimal amount of data but come up with a very accurate prediction or with an accurate detection, had to look into the existing fire danger rating systems. There have been several developed from Canada, from in the other end to Malaysia. Some algorithmic approaches to observe and then come up with a index for the probability of the fire. And this was studied and combined to develop the intelligent part of this solution. Then the other primary outcome of this research was to develop a low cost and a multimodal sensor node. Multimodal here means it's able to go for multiple sensory detections. So it's was initially targeted for developing a sensor node that is capable of measuring temperature, relative humidity, wind speed, and rain. And wind speed and rain was required external apparatus, which was to be added later on. And we had to work around some existing challenges in wireless sensor node, that this was going to be a off-site, off-grid, and away from coverage type of application. So the problems ranged from how to manage the power consumption so that you don't have to go out to the node every day to provide a new battery or so on, and how to manage the node for sleeping, waking, and maintaining the power over a long period, so that the initial idea was to have it work for at least a couple of years on a single battery. The scalability in terms of we'll have to cover large areas. We are not going to have antenna structures anywhere in the wild. So they had to communicate among themselves and find a path to tell the central node or the central server that there has been a detection, and the communication protocols that are required to develop this communication. So in this process, communication protocols, the network topology, and the error handling routines had to be designed. And finally, the findings from the fire detection indexes were combined into an algorithm. Now, the Canadian and the European standards were not directly in match with the climate conditions that we were seeing in our two countries. So we had to model the system on the Indonesian and the Malaysian models, and using techniques such as support vector machines and the K coverage algorithms, the methods could be developed for a better approach. Right. So that was the theoretical, or sort of the behind the scene part. And we come to the tangible part. We came up with the development of a sensor network and the sensor nodes. So here you can see the developed sensor node in part A, which can be simply powered off grid via a small solar cell. And if the solar cell is not viable, we can simply have an integrated battery that will allow the system to run for at least 2 years under our current calculations. And the internal workings and the basic components have been described here. And we, in this case, have included what you call a IoT device capable of Wi-Fi communication, and which is going to create what we call an ad hoc network among themselves, and including pressure sensors, gas sensors, and temperature and humidity sensors. And this node was to connect via networking using the Wi-Fi standard to a networking router that has to be established similar to the wireless node in the wild. But to simulate this and to verify the proof of concept, it was developed in the laboratory environment using a standard network switch and a server, and was connected to a main station where it would act like the base station in this case via a wired connection. And under this situation, we conducted experiments of collecting data and checking the error correction and the data connectivity and the network reliability and the other types of comparatory requirements to maintain the communication from the wireless node back to the base station. And this was tested out in the real world. This was an application in an wilderness environment. You can see the developed structures to scaling. It's allowed the system to be scaled down and then developed into multiple wireless node arrangements, which can simply be constructed at a very low cost compared to existing wireless node systems that are currently OEM. And from this, we integrated the setup into in situ. We fixed this and then collected data from the environment using the main station server, and the collected data can be seen here. Now that sensor node development and the sensor node network would be connecting to the aerial platform that would be coming around based on the wireless sensor information to locate the fire as a secondary observation. And in that case, one of the problems that was there was maintaining the stability of the drone inside the wilderness environment with the foliage and maybe the wind loads, as these are, we say, in the mountainous region. So this was experimentally evaluated in a laboratory setup, and after doing some control applications in terms of PID tuning, we could ensure the drone is stable under such fluctuating wind loads. Right. And the other part from the drone development was to ensure the drone could be able to detect the fire in its visual observation. The drone was going to carry a camera, and in this camera, it was going to observe the environment visually and then allow it to track the fire in the environment. So image processing and the subsequent path planning to go closer, observe, verify, and accurately position the fire location in the grid and to return was developed.
Over now.
Yeah, sorry. Yeah, and some modular robots were used to do the image processing and path planning part as well. Right. This is the process from the Indian team. They developed the ground robot part. And we had several visits between the two teams. The Indian team visited Sri Lanka, and the Sri Lankan team visited the Indian group and shared information and also did some field visits. And several publications came out of this, including some conference papers and 2 journal papers currently under review for publication. Thank you.
Thank you, Asita.
Thank you. That's end of category 2. So moving to category 3, food technology, energy waste, electrogeometric and oceanography. The first presentation from that is studies in surface sterilization of spices using non-thermal processes. So Dr. P.N.R.J. Amanugoda, who is from Industrial Technology Institute. Dr. Neville, shall we move to the next one and then once you get. Okay. So we'll have biopolymers, biosurfactants-based delivery micro-nano vehicles for safe delivery of food bioactives and nutraceuticals. Professor D.N. Karunaratne, University of Peradeniya, presented.
Okay. The title of our study was Biopolymer-based micro-nano vehicles for safe delivery of food bioactives and nutraceuticals. The research team, from the Sri Lankan side, the principal investigator was Professor Nidra Karunaratne. I was the co-PI and the student was Ms. Shakur. From the Indian side, the principal investigator was Professor Ramakrishna Sen. The co-PI was Dr. Chimni Hasra and the student was Ms. Varisha Farid. The aim of the project to maximize the bioactive potential of nutraceuticals and bioactive compounds via encapsulation in suitable matrices. Bioactive agents give many health benefits, but there's a problem because bioactive agents become less effective due to poor solubility, instability during food processing, degradation during storage, and degradation along the gut. But we can improve the bioavailability by increasing the solubility, stability, and absorption of the bioactive agents. We can do that through encapsulation. So what you can see on the screen is a picture of a micro or nanoparticle with encapsulated bioactive agents. You can see the wall material or the matrix in green, and in black, you can see the encapsulated material. So there are many advantages of microencapsulation. For example, encapsulation can reduce the degradation of sensitive bioactive agents. Degradation can happen due to external factors such as light, oxygen, and pH, hostile environments along the gastrointestinal tract, and processing and storage of the functional foods. Also, encapsulation facilitates a slow or controlled release of bioactive agents. Also, bioaccessibility and bioavailability can be improved through encapsulation. The specific objectives of the project were to evaluate the suitability of 3 different matrices, alginate, chickpea protein, and soy protein, in encapsulating 3 different bioactive agents, folic acid, curcumin, and hydroxycitric acid. And to physically characterize the encapsulated systems, we carried out particle size and polydispersity index analysis, zeta potential analysis, encapsulation efficiency, loading capacity. We took scanning electron microscopic images, and we did Fourier transform infrared spectroscopy. The third objective was to evaluate the storage stability, in vitro release properties, release kinetics, transport mechanisms, and bioaccessibility of encapsulated systems. And we tried to suggest the most suitable delivery system for each bioactive agent. The bioactive agents that we used, as I mentioned previously, were curcumin, folic acid, and hydroxycitric acid. The biopolymers were alginate, chickpea protein, and soybean protein. Yes, those are the biopolymers that we used. And curcumin shows many health benefits, and the major bioactivities include antioxidant activity, anti-cancer activity, and anti-diabetic activity. Folic acid also has many health benefits. It is involved in the regulation of RNA and DNA, and it is important for cell tissue and growth, cell tissue growth. It is involved in red blood cell and white blood cell production. It prevents neural tube defects, and it is involved in the renewal of skin and intestinal lining. Hydroxycitric acid is also very important. It shows many bioactivities, and it is well known for its contribution for weight loss. Project at a glance, we fabricated encapsulated systems using 3 bioactive agents and using 3 biopolymers. Then we looked at the stability of the systems. We evaluated the release properties of the system, and then we looked at the bioaccessibility. We actually wrote 5 papers based on the research that we carried out under this project. I'm going to take one study and then show the important results only. Curcumin encapsulated systems. The particle size analysis showed that we can make particles from nano to micro range. Polydispersity index studies were carried out, and zeta potential shows that the particles show incipient instability. They are sterically stable, showing incipient instability. And encapsulation efficiency was very high, and we obtained loading capacities that were only moderate. Micro nanoparticles. Now we took SEM images, and you can see that the particles were spherical, and then they are of the nano to micro range. We carried out Fourier transform infrared spectroscopy to evaluate the successful encapsulation of the bioactive agents. We show many shifts. We saw many shifts in the peaks, and we concluded that the bioactive agents were successfully encapsulated in the matrices. And stability studies, we chose two different temperatures, 4 degrees, which is the refrigerating temperature, and room temperature. And then we looked at two different, we checked the photo effect as well. We kept a group of samples in the dark, and then the other group exposed to light. And then we looked at the photo effect. What we found out was that the stability of curcumin was higher at refrigerating temperature, and it was higher in the dark conditions. And when we compare the two matrices, alginate and chickpea protein, we saw that alginate particles showed higher stability. Slow release, we actually saw that through encapsulation, we can we can facilitate slow release. I think you can see on the screen that free curcumin showed like a very high release compared to the encapsulated particles. And if we compare release and pH at pH 2 and pH 6.8, pH 2 is the gastric pH, and pH 6.8 is the intestinal pH. So when we take the alginate particles at pH 2, the release was much slower than at pH 6.8. Now this shows that alginate particles exhibit gastroprotective effect. And bioaccessibility, we can very clearly see that encapsulated particles show higher bioaccessibility than free curcumin. So we can use the encapsulated systems to enhance the bioaccessibility of curcumin. Project outputs and socioeconomic impact. We were able to fabricate nano to microparticles successfully, and we saw successful encapsulation and slow release. The stability of heat and light was improved on encapsulation, and the bioaccessibility of compounds increased on encapsulation. And also this study demonstrates that encapsulation may enhance absorption of bioactive agents in the gut, thereby increasing the bioactive potential. So publications arising from the project, we published one paper in Journal of Food Quality, and then another paper in Ceylon Journal of Science. And today we came to know that the paper on curcumin has been accepted for publication in the Journal of Food Hydrocolloids for Health. And there are two more papers undergoing revision. These are the research communications arising from the project. Suggested way forward, we would like to commercialize the commercialize this process, but actually incorporating the encapsulated systems in food or nutraceuticals will be a costly process, but the health benefits and saving for medical expenses would surpass the cost of purchase, we believe. And we would like to acknowledge the Minister of Science, Technology and Research for funding this project. And thank you.
Okay, we'll go back to Dr. Amanugoda's presentation. Studies in surface sterilization of spices using non-thermal processes.
Yes, please go to the next. Yeah. Go to the next section. No, it's go to the next one. Yes.
Okay, next presentation will be made by Dr. T.U. Aridasa from University of Moratuwa. It's a video already recorded. The topic is process development and assessment evaluation of microalgae based biofuels and value-added products. Is it ready?
Good morning. I'm Dr. Tilini Aradasa, and I will be presenting on the Indo-Lanka project titled Process Development and Assessment of Microalgae-Based Biofuels and Value-Added Products. The project was a collaboration between University of Moratuwa, Sri Lanka and Indian Institute of Technology, Delhi, India. The joint project commenced in February 2018 and was completed in December 2020. If we look at the overall budget of the project, it was 6.68 million rupees and funds received was approximately 5.5 million rupees. Next, we will have a look at the research team. The Indian research team comprised of Professor Indo-Sri Malik, Professor S.N. Naik, Professor K.K. Pant, and Dr. Vikram Singh, whereas Sri Lankan team consists of Professor Aryathala Gay and Engineer P.H.V. Nimarshana. The main aim of the project was to facilitate knowledge sharing and technology transfer between two institutes on the process development and assessment of microalgae-based biofuels and value-added products. Overall, the project aimed to develop cost-effective microalgae cultivation systems, then mathematical models and processes for synthesis of valuable bioproducts and biofuels while valorizing the residual biomass. This slide summarizes the work plan of the project. The orange arrows represent the work carried out by the Sri Lankan team. Green arrows are the work conducted by the Indian team, and the black arrows represent the work which are done by both teams. Both teams work on the development of efficient and cost-effective systems for cultivation of microalgae. The Sri Lankan side performed the extraction of high-value compounds, namely carotenoids and PUFA, and in addition, fermentation of the residual biomass was carried out to produce bioethanol. The Indian side performed biomass fractionation using hydropyrolysis, which yielded flavonoids, bio-oil, and biochar. If we take a look at the overall project objectives, first, efficient and low-cost systems for cultivation of algae were developed, and then algal biomass was synthesized as feedstock for production of food and fuel value products. Thereafter, catenoids and polyunsaturated fatty acids were extracted from algal biomass, and biofuels were produced from residual biomass. Now let us take a brief look into the work that was done under the project. The team at UOM developed a lab-scale photobioreactor array. As you can see in the photograph and the diagram, the photobioreactor array comprised of systems for illumination and aeration, and the developed photobioreactor array is a comprehensive system which can be utilized for any lab-scale experiment on microalgae. We also screened the most suitable reactors for outdoor cultivation of microalgae, Haematococcus fluvialis, which is used to produce the high-valued compound astaxanthin. As you can see in the slide, a bubble column type photobioreactor was developed to assess the feasibility of scale-up cultivation of Haematococcus fluvialis under outdoor climate conditions in Sri Lanka. The reactor was equipped with systems for control of temperature and incident light intensity at 27 degrees below 13,500 lux. We also developed an artificial neural network model to simulate the growth of Chlorella vulgaris while incorporating the effects of micronutrients. 23 culture parameters were incorporated as model inputs as shown in the slide. We studied the methods to enhance the astaxanthin accumulation in Hematococcus pluvialis, and it was determined that continuous illumination and phosphate limitation were successful in enhancing the astaxanthin accumulation up to 7% in dry cell weight. ANN model for astaxanthin production from Hematococcus pluvialis was developed under the optimum conditions determined by the models. An astaxanthin content of 3.34% dry weight was achieved in 7 days. We conducted experiments on the co-extraction of DHA and fucosanthin from marine microalgae Trisochrysis lutea. The effect of nutrient concentrations and light intensity on the productivity of DHA and fucosanthin was investigated. DHA content reached 8 to 12% of total fatty acids, while fucosanthin content reached 14 milligrams per gram of total biomass. In a biorefinery context, Trisochrysis lutea can be used to produce 14.4 milligrams per gram of fucosanthin, 11.9 milligrams per gram of DHA, then 48.1 milligrams per gram of bioethanol. If we consider the major outputs of the project, state-of-art facilities for microalgal research was developed at University of Moratuwa. Then a strong collaborative link was established between University of Moratuwa and Indian Institute of Technology, Delhi. Then knowledge was disseminated to local and international scientific community through workshops and webinars. And finally, numerous postgraduate students were trained. Sharing of knowledge and technology transfer between the institution was one of the most important aspects of the project. To this end, technical visits were conducted between the institutes. As the first visit, research students from University of Moratuwa visited Indian Institute of Technology, Delhi in May 2018. Subsequently, members of the Indian team visited University of Moratuwa in November 2018. During this visit, a workshop was conducted at University of Moratuwa under the theme Microalgae Sustainable Cellular Factories. 16 participants were selected from 96 applicants from different research and higher education institutes in Sri Lanka. The keynote speaker of the program was Professor S.N. Naik from IIT Delhi. Professor Naik delivered a keynote speech on supercritical fluid extraction, an efficient way to extract high-value products from microalgae. And Dr. Megha Mathur from IIT Delhi conducted a lecture on microalgae bioprocessing for a sustainable future. The workshop comprised of laboratory sessions which introduced the concepts of microalgae isolation, cultivation, then product extraction and analysis techniques to the participants. In addition, interactive discussions were held with the participation of Professor Naik and Dr. Megha. Here you can see all the participants and the organizers of the workshop. A webinar titled Global Trends in Algal Research, Environmental Restoration, Biomass Valorization and Sustainability was organized under the project and was held on 8th and 9th March 2021. Over 290 participants were in attendance and 17 renowned keynote speakers graced the event with their expertise in algal research. Now let us take a brief look into the publications that were a result of the project. Several joint publications were published and communicated under the framework of the project. This included journal articles on biotechnology reports, Biochemical Engineering Journal, Algal Research, Bioresearch Technology, and a joint book chapter in Algae and Aquatic Macrophytes in Cities, Bioremediation, Biomass, Biofuels, and Bioproducts was also communicated. The Sri Lankan team also published a review article in Food Chemistry and a journal article in the Journal of Biotechnology. Overall, there were 6 journal articles and 1 book chapter with Sri Lankan affiliation. Also, the Indian team presented a poster as a joint proceeding in the Bioprocess India 2018 International Conference. The Sri Lankan team also published Scopus indexed full conference papers in Mercon 2018 and Indicon 2020. Furthermore, the research findings were communicated as poster presentations by the Sri Lankan team. As for the suggested way forward, there are plans to formalize collaborations between IIT Delhi and University of Moratuwa via an MOU. And furthermore, the results of the studies conducted under the project can be applied in pilot scale to gauge the techno-economic feasibility. This will enable us to identify the feasibility of producing export-oriented algal bioproducts in industrial scale. Most importantly, I would like to thank the students who worked tirelessly to achieve the project objectives. On behalf of the IIT Delhi and University of Moratuwa, I would like to take this opportunity to thank the Indian team, which included Dr. Poonam Chowdhury, Dr. Megha Mathur, Nidhi Hans, Farah Naz, as well as the Sri Lankan team, which included Vinod Lianarachi, Malik Premaratne, and Manjita Ranmani. So that's the end of our presentation, and thank you very much for your attention.
Thank you. That's a video presentation, but I think one member of the team is here. Next, we will have reconfigurable architecture for solar photovoltaic microgrid systems. Oh, sorry.
Sorry for the delay. That is obvious in some times. My project is studies in surface sterilization of spices using non-thermal process. And project teams from the ITI side, I myself and Mrs. Ramya Pittipanarachi, she's a co-I of the project. She's a research scientist now. She's retired now. And team member 1, Ms. Gayatri Silva, research engineer, food technology section. Now she's carrying out a PhD program with the University of Moratuwa. Professor Ajit Dallis and Professor Sanja, the internal supervisors, and I myself as the external supervisor. And there's another team member, but he at the moment he's leave the ITI, Mr. Buddhika Veer Singh, research assistant, food technology section. From the Institute of Chemical Technology, ICT Mumbai side, Professor U.S. Annapuri, head of Department Wood Engineering Technology, Institute of Chemical Technology, Mumbai, India. And now he is the Director, Institute of Chemical Technology, ICT, Marthoda Campus, Jalna in India. And the co-PEI, Professor R.R. Senior Professor R.R. Deshamukh, Department of Physics, and now she's the registrar. Maybe he will promoted to Vice Chancellor of the ICT Mumbai, India. And the project duration was 2 years from 2017 October to 2019 October, but there was officially 6 months extension. But we were carried out the project up to last year because of the PhD programs. total allocation from the ministry side is 11.8 million, but we spent from ITI side 5.8 million, but from the from the ministry side also they spent some money, almost 6.5 million. Remaining part, there was a problem because of the country situation. Coming to the problem, because we know that Sri Lanka and India are the most spices manufacturing countries and exporters. So we focused that to select the spices to address one specific problem in the spice industry under the area of sterilization. As you know that there are a lot more sterilization techniques that belong to steam sterilization, but when we applied steam sterilization, it may damage to the product because spices are very sensitive product. So it may damage to the volatile content, color, appearance, and so many aspects. So there's emerging techniques under the non-thermal plasma, non-thermal techniques. There are almost more than 12 non-thermal sterilization techniques. We choose that UV technology and cold plasma as the non-thermal sterilization techniques to apply under this project. And the drawback of these two techniques is that these are surface sterilization techniques. So it's one of a barrier. And when we apply this technique into the spices, you know that the spices are very small particles, then there'll be shadow effect. So if there's low microbial content only, we can apply these techniques. So specific objectives was optimize the cold plasma UV process parameters for effective killing of the microbial population on the surface of the selected spices, evaluation of microbial inaction from surface of the selected spices, effect of cold plasma and UV treatment on physicochemical parameters, sensory and qualities and shelf life of the selected spices, and whether if we can combine these two technologies and go for a more advanced techniques. We come to the key findings under the cold plasma technology. We import this atmospheric pressure cold plasma gliding or discharge from Mumbai, India, and we optimize it to do the sterilization techniques. And these are, we put some results by using gliding or pressure cold plasma discharge treater. You can see that there's a relative level of log reduction by application of the gliding as plasma discharge, and also with help of Professor Ajit Alvis, and you could know that there is one of vacuum cold plasma mass innovation center. So we went there and we have done several trials. We put some significant results here. You can understand that by using vacuum cold plasma also we were able to reduce the log reduction in black peppercorn for the APC 0.88 with 15 minutes treatment, and Eastern mole 3.66 log reduction with 15-minute treatment. But in case of paper powder, there's a little bit reduction of the increment of the time. We were able to have this result 0.98 log reduction and for the APC and 4 log reduction for the Eastern mole and chili powder. Also obvious results we obtain. But the things that when we apply longer treatment times, there'll be tendency for the increment of the temperature, and that will cause to loss of moisture, loss of volatiles, color degradation, changes in chemical structures of the surface. So we are trying to minimize the time taken for this kind of surface sterilization techniques. This is the radio frequency powered rotary dielectric barrier discharge. After that, after trial of gliding discharge and vacuum plasma discharge in ITI laboratory, we made this and Ms. Gayatri for her PhD program with support from Morocco Institute also. We developed this rotary radio frequency powered rotary dielectric barrier discharge reactor. And also optimization is going in progress, and we have an idea of to scale up from this is the laboratory scale one, and we want to go for the industrial pilot scale one. Thereafter, if there's a possibility, go with the industrial scale, because in the fabric trade, there's a 10-meter width plasma probe applied in the industrial fabric industry. So there's a possibility if we can find out what are the barriers and fine tune the equipment. And the key find is UV technology, and we found that UV technology has the ability to kill the microbial present in the black pepper, seeds, black pepper, powder, and cinnamon, and other spices also. So we were able to develop UV rotary drum and UV canopy system, and thereby we were able to increase the effectiveness of the UV treatment by 10 to 15, because in the existing sterilizers, they showed that the efficiency is around 35 to 40%. So altogether, our treatment, we were able to increase the efficiency time up to 55 to 60%. Sometimes, in some cases, more than 75%. And these are some fungal and bacterial isolation under the UV technology study carried out by the Buddhika Veersinghe, my RA, because earlier he thought to register for his MPhil program, but with some reasons he couldn't able to do that. And we combine the cold plasma with UV technology, and also we combine only UV canopy, but there's a possibility to combine UV rotary drum with the UV canopy and gliding and discharge cold plasma system. We have proven and these are the isolation of bacteria and fungi under the cold plasma treatment, and have the possibility of getting accession numbers for Sri Lankan black pepper and chili powder. And these are project output, established facility post by peak contribution by APCPGAD and vacuum plasma at Mass Innovation Private Limited, optimized for decontamination, and UV rotary drum and canopy type, and combined APGT and UV canopy system was optimized for decontamination. And lab scale atmospheric pressure rotary DBD cold plasma we have designed and scaling up for the optimization and morphological and genetic identification microbes around 29 in selected spices, delivering of services for cold plasma parameters to industry. We have initiated and measured to apply findings to industry. We have a discussion with Vijay Industry and small spice companies to deliver this technology and initiate and near to completion of PhD program under the cold plasma, and design MPhil PhD program under UV technologies and spice microbiology remaining. And we have an idea to initiate. There's a chance at once to start this and complete the study. And we have done undergraduate studies 5, invited internships I have delivered around 6 in the India, advisory committee member of the ICAPS cold plasma symposium organized by the Coimbatore, India, 2020, 21, 23, and I am one of the reviewer of the IEEE transaction on plasma science. From this project only, I got this opportunity, and we won the best project with national significant output organized by the ITI Symposium 2021, and we got to know that we have nominated for the CARP awarded for this year. And these are the publication we have done, almost done 2 publication, ready for submission 2 publication, and in preparation 2 publications, and 14 communications. And these are the suggested way forward, pilot and rotary type vacuum plasma development. And this pin to plate plasma in India, we have idea to, this is a very one of a very useful instrument. So we have idea to collaborate with India and complete the study. And finally, thanks to Ministry of Education, Skill Development and Vocational Education, Research and Innovation Ministry, and State Ministry of Technology and Research, Department of Science of India, and ITI and ICT Mumbai and NS to organize this event. And to the panel members and my previous panel members, evaluation members in the committee, they gave full support to direct us to obtain best results. Thank you, sir. Thank you.
Thank you, Dr. Amanugoda. We'll move on to the next paper. I think Professor KTMU Hemapala from University of Moratuwa. Reconfigurable architecture for solar photovoltaic microgrid systems.
Have a nice day to you all. I am Himali Lakshika. Today I'm going to present our research project. It is design and development of new reconfigurable architecture for solar PV microgrid. This research was done by the collaboration work from India and Sri Lanka. From Sri Lanka, our professor Hemapala was the investigator of this project, and from India, Dr. Saruna guided us for this project. This is the outline I will be going through, and this is the research team. As I previously mentioned, I also work as a research assistant for my master's degree, and Kallahan Burlasa and also Hasita Eranga contributed for this research from as our Sri Lankan project group. From the background, it is a electrical system and solar PV system based research. So I think it will not be familiar sometimes. I am telling about the technical words, but you all know what are the solar PV systems. But in the current technology, our solar PV systems cannot withstand if there is a power failure in the utility grid, or if there is a fault in the utility grid. So our current solar PV system cannot withstand and generate solar generation from our solar PV system. So actually it is a problem for our solar PV systems. So we needed that connecting architecture, and we should be rearrange that architect to connect the solar PV system to the utility grid. In the current solar PV systems have an inverter. It is normally voltage source inverter, but in here we are talking about Z source inverter. Normal voltage control voltage source inverters, we have to keep dead band, because normally that inverter, we short circuit that inverter sometimes, but it will be caused to failure that inverter. So that's why our solar PV system had shorter lifespan. Even though we predicted it is 20, most of time our inverter will fail around 10 years. So with that sense, we observed what are the solar PV inverters we can use. In the research background, we found there is another type of inverter called Z-source inverter. It can withstand this failure system. So with that sense, we focus about Z-source inverter in this research. And also there is a requirement of reactive power demand in our utility networks, because in our utility networks, there are under voltage situation within the, when there are heavy loads in the distribution network. Not only that, when we connect solar PV system into the distribution network, there is a voltage rise in our distribution network. Those are the limitations to connect solar PV system into our distribution network. So we need reactive power support for our distribution network. So that can be achieved by providing reactive power control or voltage support control from solar PV systems. So that is a new trend with these solar PV systems. Not only that, as I previously mentioned, our solar PV systems, they are just grid feeding inverters. They cannot withstand when there is a grid failure. So that's the point we needed. Islanded mode can microgrids that can be work as a islanded mode. And with that sense, you all know our solar PV system cannot connect to the solar utility grid as a single architecture or single configuration. So it should be have several configuration to support this our utility grid. So it should be have the function of reconfigurability. So as an introduction, this is the concept of microgrid. It is a group of generators and loads, and also there is a complete boundary. It can also withstand as a separate power system. And this is the definition of the reconfigurable system. Reconfigurable system can work for the separate objective, and it can change its configuration to achieve different objectives. So this is about this Zerzov's inverter. As I said, there are so many reasons to fail the inverter. One of them, our Zerzov's inverter can help us to increase the lifetime of the inverter. So, as I mentioned earlier, this is our main objective, to develop a reconfigurable architecture with these solar PV systems. At a glance, this is what we proposed. This is our proposed architecture, microgrid architecture, and we developed a control algorithm for this solar PV systems to work with. And also we could implement this Z-source inverter at laboratory. The major outcomes of this project, we could actually propose a reconfigurable model. It was a simulation model. We could reconfigure that solar PV system in 3 different configurations, and also we could implement the Z-source inverter prototype. And also we could publish 3 international conference papers and also journal papers. This is what I propose as the reconfigurable architecture. As I previously said, it can work in 3 different modes. One is current controlling mode. It is the default mode, and the next is voltage frequency mode. At there, our system can support the distribution network by providing reactive power. The third configuration is voltage frequency control mode. At there, if there is a grid failure, our network can tolerate that, provide power to our power system. In addition to that, we propose a communication link. In the international standards also, now this communication link is requested. So with that communication, we can select what is the mode of operation and also what is the operation we needed. So utility grid and also home microgrid can communicate. This is the prototype implementation we have done. Those are the publications. And as I mentioned previously in my background, this is the conclusion. With this project, we can, our consumer also get benefits, and also utility give the benefit. And also as a national, we all can get benefits from this architecture if we can implement this type of architecture. Because with the microgrid reconfiguration and islanded mode, we can support our solar PV systems to our loads also, and also critical loads. This was the budget allocation, and this is the financial progress. As a future, we can actually, the main barrier we had was Z-source inverters are not commercially available. Actually, it is in the research background. There are some components needed to develop the solar Z-source inverters at the commercial point of view. So our IGBT, some part of inverters should be developed furthermore to use this utilization of this Z-source inverter actually, but we can use this architecture for the existing solar PV systems. It is also now in the practice in other countries, but not in Sri Lanka. So it also included into the international standards. So we hope in the future it will come in our practice, it will become to our practice, and our network can absorb this kind of solar PV systems. Thank you.
Thank you very much. Now we are going to have another online presentation, this time from University of Peradeniya. That will be made by Professor M. Fernando. The is protection of heritage monuments and landmarks of national and international importance in India and in Sri Lanka due to direct lightning strikes, traditional and scientific methods. Professor Fernando.
Am I audible? Am I audible?
Professor Fernando, you can start now.
Yeah. Am I audible? Okay. Yeah. Good afternoon. I am Manjula Fernando. The title is about, it's a lengthy title about the protection of heritage monuments, which includes mainly stupas in Sri Lanka and Kovils, Hindu Kovils, church, etc. in India part. And it covers something like the effect of the direct lightning strike, traditional and scientific methods. So the project team, from Sri Lanka point of view, myself and Dr. Sarath Kumar Kopiai, and from India, the collaborative institute is Vellore Institute of Technology. From the elect department, Professor Venkatesh, and from the civil department, Professor Tirmalani. And there was no dedicated student from the either side, India and Sri Lanka, to conduct this research. But there are a couple of students who did undergraduate project under our supervision, etc. So since this project was some kind of a multidisciplinary nature, so we have included several team members and some advisors. So Dr. Jayaratna from Department of Archaeology, because it's related to the heritage monuments, from University of Peradeniya, Professor Susantha from mathematics, some mathematics modeling also there, Professor Mahendra Fernando from University of Colombo. Apart from that, as advisors, we have included Professor Vernon Kure from University of okay. Right. Okay. Okay. As the advisors, Professor Vernon Kure from Uppsala University of Uppsala, Sweden, as expert in lightning, and Professor MP Ranavira as experts in finite element methods, etc. So the project started in 2017 December for 3 years, and the budget was actually 3 million from Sri Lankan side, but we have utilized slightly less than 1 million rupees due to some of the practical situations in the country. Just to give an overview about, because there are two keywords, one is the lightning, the other one is the protection against the heritage monuments. So when you consider about the lightning, actually we have to consider about basically the cloud to ground lightning, and there can be 4 different options. The frequent one is the negative lightning starting initiating from the cloud to ground, and there can be positive thing. And if you consider about some, I mean, tall structures like heritage monuments, so we have to consider about the ground initiated lightning, so they can be also positive and negative. So what will happen is the cloud, usually the charges are accumulated and the electric field intensity increases. So as a result of that, the step leader, the electric field goes down as a step leader. And so usually the distance between the ground and the cloud is around 2 kilometers. When the step leader reach near the ground, so the ground will oppositely charge, and at some point there can be some kind of a bridging between the the opposite charges, and it will start the return stroke as the lightning. So based on these phenomena, different lightning protection methods are established. So when we consider about the heritage monuments, so there are different heritage monuments. The main concentration like the stupa, and usually we know that almost all the monuments they are protected against the direct lightning by installing the lightning protection system. Going further down. Okay, the objectives as listed in the project proposal, there are so many objectives listed under the project proposal, but so we have mainly considered most of them, but I will concentrate some of the major things. For example, lightning protection system, the standard way based on the geometrical and electrogeometrical models based on the IEC standard against especially the under the heritage monuments and structures. So usually we have considered stupa, kovils, church, etc., and statues like Aukana, etc. But here what I mainly concentrate on the major heritage stupas in Sri Lanka and standard and non-standard methods related to that. So to give an overview about the project at a glance, so this is some kind of a pictorial way how to present the different protective measures, protective systems based on different criteria, and their explanations are in both sides. Here, left-hand side, it has the standard ways. The other one is the that is also standard, but some kind of a non-standard way. The one of the method is the protective angle method. That is the simplest way. So based on whatever the area, so we draw some kind of a cone, and the coverage under that is protected. So the second criteria is the rolling spear method, that is based on, so the first one is based on the geometrical model because it's mainly depend on the geometry. The second one is the electrogeometrical model, we call the rolling spear method. So here it's the lightning leader, and this is the lightning tip. And this is the structure, and in between these two, there is a striking distance. So depending on the light intensity, so the striking distance can be calculated based on different formulas proposed by different scientists. And again, it different depend on the lightning current as well. So the typical lightning current can go up to 200 kiloamperes, and the typical level is like 30 kiloamperes. So different protection levels are also proposed in whatever the standard. So we have tested those also. So the rolling spear method, that is another one, the electrogeometric model. So based on the touching area, we can see that which parts are vulnerable to the lightning. So another extension to the rolling spear method is the modified rolling spear method, that is specifically for the ground, that means the structure initiated lightning. So this is another cone like that. So apart from this standard methods, so we have concentrated on the finite element based method based on the console computer software. And So that will they are not only the geometry and the electric model actually for the lightning. So we have considered about the ground condition. That means the ground conductivity, etc. Those things. And based on that, the electric field distribution, potential distribution, they were calculated. And based on whatever the human interaction was also proposed. And these are some of the selected stupa out of, 5 main stupas in Sri Lanka and 2 main Lord Buddha statues in Sri Lanka. India also, they have considered 2 povils and 2 churches, etc. So in our study, what I have presented here is the main 3 giant stupa in Anuradhapura era, Jetavanarame, Abegiri and Ruwan Velisaya, and in Polonnaruwa, Rankotvera and Kirivera. The difference is they are very tall, and some of the structure has some disturbance, and the total height is not the actual one. On the other hand, from the Polonnaruwa part, these are quite smaller in scale, and also the contrary to the other 3, they don't have any installed lightning protection system. But for these 3 cases, there are lightning protection systems installed on their top of the pinnacle. And so there, even in the media, there are some news about some lightning effects for different monuments in Sri Lanka, but from the archaeological department, they are not officially disclosing that none of the whatever the monuments are struck by lightning. So I'm not going into detail, but what we have done was the investigation of the lightning protection system in all cases, including povils, churches in India also by them, under different methods, electrogeometric, geometric method, and the finite element based methods. And these are some kind of test results based on different equations. And these are some kind of the largest current, lightning current, which can happen, which can strike to these are some estimated values to those monuments. And the probability, it is for the same probability. When we consider about 132, that means it is the probability for 132 or above is it's only 2-3% of the probability. So it is very less probable. Okay, I will quickly go through them. So by applying the rolling sphere method, so we can find out whatever the one, and this is the finite element modeling one. And we have gone through about 2 kilometer object, and these are the field distribution thing. And the human interaction methods. So we have done about 5 different human interaction methods, and these are some of the results. And the outcome of the publication, so the main publication is IEEE Access. So all the information are available in the IEEE Access, the main findings, apart from that sustainability journal. And we have, published several conference papers and some popular journal papers. And apart from that, we have conducted tutorial in the 14th IEEE ICIS International Conference, which was held in 2019. The title is "There are software tools and techniques for lightning protection application." So as the PI, I have conducted about the lightning protected protection basis, and from PI from India, Professor Venkatesh, he has conducted part of the workshop, and we have invited Dr. Chandi Mekanayaka from University of Queensland. And these are the project summary and the way forward. The tested all stupas are prone to lightning, and different standard, non-standard methods can give different risk levels. So that is the thing. And with different standard, we can get different results. Then with finite element method, possibility of initiation of upward leader, so possible lightning. Then heights of the present stupas, they are lower compared to the ancient heights. So due to the renovation, as a result of that, lightning risk has increased. So that is one of the finding of that. Then by comparison of geometrical, electrogeometrical, finite element based modeling, we can conduct a very comprehensive protection measures and do a thorough one. So those are the things what I want to present in this symposium. Thank you very much.
Thank you, Professor Fernando. We'll move on to the next one, again, online presentation. Professor Malavi Arachi from University of Peradeniya, probing the provenance and distribution of heavy mineral based deposits in coastal areas of southern India and Sri Lanka. A new multi-mineral proxy approach to understand source to sink link. It's online again. Professor Malavi Arachi. Professor.
Good afternoon, sir. I hope I'm audible to all of you.
10 minutes, please.
Yeah, sure. Let me start the presentation. My topic is probing the provenance and distribution of heavy mineral placer deposits in coastal areas of southern India and Sri Lanka. A new mineral proxy approach to understand source to sink link. This is the project name and number, and we started the project in 2017 and we completed it in 2020, but we had to move on a few months for financial matters to be clarified. But anyway, the main tasks were completed within 3 years. And this is the project team, principal investigator, myself, and co-investigator, co-PI, Dr. P.L. Dharmapriya from the Department of Geology, and a collaborator because we had have to access some equipment in NIFS Kandy, Professor N.D. Subasinghe. And there are 2 students, AMGK Vijayaratna, UGAMP Abhayavardhana, MPhil students. From the Indian side, principal investigator is Professor Sajeev Krishnan from the Center of Earth Science, Indian Institute of Science, Bangalore. And the PhD student is Miss PG Athira. The general aim of the project is to investigate the source of the placer mineral deposits by comparing the petrology, geochemical characteristics, and the radiometric ages of placer mineral deposits and various lithologies, rock types in the study area. Specific objectives of this project was to characterize hyperspectral using hyperspectral imaging, and to map the distribution of different placer deposits, different mineral deposits along the coastal areas of southern India and Sri Lanka, and quantification of coastal and placer mineral deposits, their age calculation, and also to find out the fertility of rare earth elements potential to make it commercially viable or not, to understand that, and to find the tectonic significance of these heavy mineral deposits in relation to southern Indian and Sri Lankan context, coastal context, and to develop necessary manpower and training in the related field, and also to do joint research publications in the area of geology, sedimentology, geochemistry, geochronology, and tectonics. Project at a glance, we started the project in the first year mainly with the fieldwork, sample preparations, and then we started in the second year mainly on rare earth elements and chemical analysis, and the third year mainly on isotopic analysis for geochronology. And in accomplishing this, first year we had this systematic sample collection where we collected samples for sieved and wet sieving, dry sieving and all for heavy mineral separation. Heavy mineral means garnet, ilmenite, rutile, zircon, like economically important minerals. And then we prepared grain mounts, that means grain slides with selected mineral grains to understand their chemistry and their morphologies. And we started geochronological studies also by separating zircon grains, because zircon is very resistant mineral and which is the one used for determining the age of the earth also. And then we were planning to do mapping the place deposits using hyperspectral remote sensing technique and also using air photographs and satellite images. But unfortunately, the Indian Space Institute in Trivandrum did not allow us to access this hyperspectral remote sensing method. And also for the Indian team, it was inaccessible. So therefore, we had to give up that task in this project. Background for the study, as you all know, heavy minerals, that means minerals which have high specific gravity, is very essential, very important for understanding the potential of a sand deposit to make it commercially valuable. And also placer deposits, that means the deposited minerals around the coast should be an important source for mineral exploration because they are available in large quantities because of accumulation due to oceanic currents. And also significant amounts of these economic minerals definitely uplift the economic the potential of the country, and therefore it's very important to explore the coastal areas of Sri Lanka, because we had some known deposits in Pulmudi areas, northeastern side, but there has been no detailed studies from southwestern coastal areas of Sri Lanka. That's why we initiated this study with Southeast Indian coast as well. So these are the sample locations starting from Hambantota, Godawai, Ambalantota, Mathura, Dondara, and we covered all those areas. And also we covered, for comparison purpose, northeastern coastal sites as well, which is not shown in this table. So this is these are some field photos from the sample collections. You can see our Indian collaborator, Professor Sajeev Krishnan, is also here. So the Indian team and Sri Lankan team together did sample collection in Sri Lanka, but unfortunately due to COVID pandemic situation, we could not visit the India for field work. So this project, although was a joint study, we were unable to do field work in India. So these are some representative slides of the separated mineral grains. You can see these reddish grains are garnet, and the brownish ones are rutile. And also you can see some colorless to yellowish, pale yellow grains as zircons. So these type of mineral varieties are abundant in the coastal areas. The only thing is we have to see their abundances individually to understand the economic potential. We did particle size analysis of these samples using the sieve analysis, and we found that the predominant grain size was ranging from 180 micrometer to 300 micrometer in these areas. And also with respect to the sorting of these sediments, these samples, because a lot of factors are there, wind and also the oceanic currents, and mainly the medium-sized grains of zircon and garnet were well sorted in these sample areas. That means they distribute in consistent grain sizes in the studied area. So that is an important observation. And also, if we see the abundances of these grains or the model percentages of these heavy minerals, Hambantota, we can see the garnet is predominant. And also this Godawaya, we can see quartz is predominant, but it is not an important mineral in an economic context. So therefore, but we have opaque minerals, for example, some ilmenite, some magnetite like oxides, iron oxides, they are there in almost all these areas. And if you consider the Beruala, you can see monazite is the dominant mineral, which is a lanthanum cerium phosphate, rare earth element phosphate mineral. So that is also very important for our economic potential to make it economically important grain mineral. And in Kalutara, again, opaques are dominant. That means iron-rich minerals are there, but still for iron extraction, we can utilize them in the future. Some important observations, as I mentioned before, these model percentages, garnet, ilmenite, magnetite mark the highest model percentages, and the Hambantota is the garnet dominant area. And also in significant amounts, we can find ilmenite and magnetite together as well. And zircon and monazite indicate location-specific concentrations, for example, Beruala specifically. So they, and also the southern coast, some Godawai area, where we could find some concentrations of these zircon and monazite. And we did some age dating. Actually, this part was mainly done by the Indian side. There you can see some separated zircon grains. These are the zircon grains, and we did, this is the cathodoluminescence images of those zircons, and we did age dating using uranium and lead isotopic method, using laser ICP-MS at the Chinese Academy of Science. And we have obtained ages like 2,739 million years. So these are the ages. For example, this grain 1,844, and this one is pretty younger, 484 million years. So likewise, we could obtain their ages, very old ages, and spanning from Gondwana to Precambrian times. So this is the distribution of these grains. So these are not any recent grains and very ancient grains. So we investigated the rare earth element distribution as well, and we can see, you can see clearly heavy rare earth elements here in this right-hand side of this diagram. Hambantota samples showing enrichments, and Panadura to Kalutara also show some enrichment of heavy rare earth elements as well as light rare earth elements like lanthanum cerium. But relative to Hambantota area, these heavy rare earth elements are at moderate composition, moderate abundance in Panadura to Kalutara. In Gol and Mathara, less concentrations of heavy rare earth elements are there. This is heavy rare earth element abundances with respect to different sampling locations. Only representative sample locations are indicated here.
Time is up.
Okay, and social economic impact is the mainly rare earth element extraction, and also the identification of heavy mineral deposits and their significance. And we have published 5 papers in international journals, high-ranked journals with impact factor greater than 4. And also we have conference and symposium abstracts, quite a large number of abstracts we could publish, and we have 1 manuscript in review, and also another manuscript is under preparation. So this one is an Indian collaborators' main manuscript, which is now in review. And the way forward is resource estimation is the main important thing for these mineral deposits, and also as suggested by our reviewers during some of our previous discussions, carrying out airborne magnetic survey around the coast is also very important. So this is our findings, and I would like to thank the Ministry of Science and Technology and also the Department of Science and Technology, India, for funding, and also the review panel for constructive comments throughout the project. And also my special acknowledgements are due to former directors, Ms. Himalia Taudage and also Mr. Karyappa, and their management assistants, Ms. Padmini Damukol and Ms. Dayantini, who did remarkable work to make this project a smooth one. And also the present staff of the Ministry of Science and Technology is highly acknowledged. Thank you very much.
Thank you. Right, that brings to an end 15 presentations that's scheduled for the day. So the next item is actually dialogue on the way forward. The organization tells unless we have a dialogue on the way forward, we do not have the lunch. So how do we do this? So we had, I suppose, when you listen to, there's so much of potential and so much variety. That's right, a significant number of presentation. So how do we move on this? What's next? All the projects, there seems to have been, not all the money has been dispersed. And even when there's rupee 180.63 has been recovered back, right? Though you didn't get 2 million out of that, yeah. And then there were suggestions like, okay, have the Pentagon all in one place in multiple areas. So can we take this, can we look at it this way? So we'll take the category 1, plant-based medicine, and just get to know a few other areas, few things that maybe you can be specific as, okay, what we should do differently or what we should be doing next, and the requirements, some key things specifically, maybe that.
Yeah. Since I was there for previous POC also, one of the things once Indian team came here, one of the things they expect was there should be active collaboration between both countries. You see, if you can do your research on your own, then you don't need that collaboration. They kept on saying that we have to identify a project where both can benefit out of the project. Now, for example, the last project that we have to collaborate, otherwise they can't get those results. So like there are some projects when I listen to them, active collaboration taken place. So that's very important. So going forward also, we must identify areas where certain expertise we don't have, India has, certainly they don't have, we have. So that's our project, high priority will be given. So I think for the next POC, government ministry has to put some proposal. So now itself, you must identify parties on that side, not wait to a last minute, and see what sort of project both should collaborate. Ensure eventually when you look at the active collaboration, one of course the publication, that should be joint publication. You can publish on your own, then you don't need that collaboration. So joint publication is one way of seeing whether there is an active collaboration. And one of the project I noticed that they have very good joint workshop seminars, that Moroto one, where they have invited experts here. So they have gone beyond the project also. So a lot of training opportunities are there. So that is one webinar. So that's a very good active collaboration. And someone mentioned that there's long life partnership. That means I didn't know whether they got married or something like that. I don't know. So that's sort of long life partnership are important because even if you don't have funds from this, you have active collaboration between those two institutions. That's also a very good way of indicating the Indian side that this sort of a partnership is very, very important. So I feel that I think a lot of projects I noticed that quite a lot of good partnership have taken place. But if you are going forward, I think project identity can become important. And he also was there earlier.
It's true in general, as I said earlier, that output is very good. Almost 40 research papers in cited journals and so many conference papers, maybe 100, I think. And there were so many visits. Both teams have come to. Indians have come to Sri Lanka, Sri Lankans have gone to India, and some juniors were trained there, as can understand. That's very good. And I think in future, we have to select problems that are relevant to both countries. Of course, you can have some basic theoretical projects, that's very good. Out of 30–40% can be devoted for basic sciences, that's very good. But most of the projects should be on problems that are relevant to both countries, Sri Lanka as well as India. So we have to, when we are thinking about the future, we have to go along that line, especially in view of the current problems that they have in Sri Lanka. We have to devote science and technology to solve our own problems and the countries around.
Thank you. By listening to the overall project outputs and results, I observe, as highlighted by the secretary in this morning, if we focus or look at in terms of benefits filtered down to the society or the relevant stakeholders, I observe there are some research outputs which has commercial potential, field level application. So to ensure field level application and realize socioeconomic benefit, I can highlight some reconfigurable framework for grid that related solar connecting to the grid, because national policies towards going to towards the renewable from fossil fuel to renewable. So mainly maybe solar, being a tropical country. Therefore, these kind of applications may be highly applicable and relevance timely for the national context. Therefore, I suggest better if you look at research commercialization point of view. One of the leading universities in the world is Stanford University. Stanford says if you invest $1 for R&D project, you have to invest $100 for commercialization output. So therefore, I suggest better in the next round, parallel to the next round call, if the ministry and all together could look at some allocating some funds for furthering these project outputs towards commercialization, at least to push technology readiness level from value chain to commercial ready version, maybe with some scaling up studies. Of course, before investing, we may need to do some techno-economic feasibilities to ensure that at the end of the day, the second, money invested in second time will ensure socio-economic benefit. Ended up with having economic impact. That is my observation from National Science Foundation point of view. In addition to that, research commercialization may not, not easier than said. We need more where intellectual property sharing or involvement, entrepreneurship and all. NSF is in a position to facilitate that process, of course, in terms of intellectual property related agreements and everything in that sense, in the commercialization process. Thank you very much.
Thank you, sir. I also want to thank and congratulate all the researchers presented today. And as Dr. Shanta said that NSF as a funding organization, we are behind these researchers. We know that on research into commercialization is not a single step, it's a process. So we have to do more and more research to increase the technology readiness level. But we have seen that there is a one project out of 17 project has already commercialized. So we can we have seen that out of 17 projects, there are some projects they have a good potential outputs, and we can think the some good product or process or some services in future from that research. So as Dr. Shant said, we NSF like to support those projects in our future activities that they like to work with us. And again, thank you all. Thank you.
Well, those who are joining and also can open up and say, because we have online. But adding to what Dr. Mubarak said, that in the last presentation, we had the situation, now you said about the Indians stressed about the participation together, but that hyperspectral one was, they said, okay, you don't have access. Right, so it's in the program, project planning, but then Sanjeev, it's from Professor Malaviarache. Yeah. But they have said no. Even, yeah. No, that's remote sensing. This is remote sensing. They have the remote sensing research station, but they are not open it up to the researcher. Both, not only Sri Lankan, but Indian as well. So it's probably in the project planning. Maybe the discussion one can highlight. So the project was planned, that party was involved, but then finally they need to open up. So hyperspectral, this is a. Yes, yeah. Okay. Very quickly, going through the groups, anyway, anything you can add on to something? Yeah.
Previous review, I can remember some of the projects, they said there was no support from the Indian side. I think from the Nidras one. I see Nidra. I can remember there was an issue because if you can indicate those issues, then going forward, we can minimize those things. There was some problem on the Indian side. There was no collaboration. Yes. In that particular, do you know the person beforehand when you collaborated, or just how did you pick the person, party, institution? Okay.
They want to collaborate, sir.
Okay. I can remember last time there were a lot of issues there in that particular project. There was no collaboration, but you all did your own work without.
That's right, sir. Yeah.
But it's important to plan the project that has been very well.
Even I think you also had some problem now in collaboration on the Indian side. Yes, this nano.
But they sent the extract, so we did the content line. And we were supposed to write our extract to for another experiment, but that art also they couldn't do it because of the lack of. They were almost for more than a year. I think they could not access labs, and that was the time that there was very heavy. The area was very heavily affected. They couldn't access the labs basically. Now they have started doing so. We have not asked them to send nanocomposites and so on because we don't have money to work on them. So we have just closed the grant. So we do not know, if we find any money, but other than the project, in general, I want to say now that I do not know, this has to be at a higher level. Now that this research, science and education all are supposed to be under the Ministry of Education. And I was happy to hear the comments made by the Secretary Education. But at the ground level, it is not realized. It is not percolating in the sense now the universities are not getting even the salaries. Now, IBMBB is like my institute. The only money that we have got is 70% of the salaries. They say you should generate funds and you should be self-sufficient. For next year, nothing. So at the moment, the staff are involved in trying to do various courses and generate funds to keep the place, prevent the place closing. So, it's very difficult to, even what you have to concentrate on research and do. And whatever the research which is going on now, like we have got some collaborations with private sector and so on, that is what is going on. So, you can say that research and development, and then if you do things to the extent that the existing places close, then you will have to start from A, B, and C in a few years' time. So I do not know. I wish the Secretary of Education was here to look into those things. I know that the country is having a major economic issue, but one of the things that the UGC has proposed to the Treasury is that none of the postgraduate institutes will be given any money from the government. So,
That's been taken up as a different forum,
yeah, I know, but
not only for everyone.
everyone. So, I mean, if whenever you all have any feasibility of discussing those with those people, I think, because we will be talking here and presenting everything and going, and then we can't go forward unless those things get sorted out. Yeah, different way forward.
Anyway, I don't just have a comment on this. Look, when we look at the presentations and the research, we discussed how many papers, conference publications, and so on. Only one presentation had a patent mentioned. And but in couple, there were one of, okay, come, there's a ready place. We will happy to support commercialization. That's President Sanayaka. Dr. Neville, you had on the patent. And then President Edras had, there's so much of potential with this microencapsulation, and then the issue of turning around the current, maybe the nutrition issue. I mean, the potential is there. Is there a way of looking at the way we do research in a much more collaborative way, and having, of course, the ministry, when they disburse funds, to have a little bit more flexibility? And when you're looking at the flexibility in terms of looking at the results to be realized, rather than looking at the financial process to be kept sacred. The issue is like, I mean, like you had the green nano, the particles production. Can you reach out to like Slintech to be very quickly to see that, okay, why don't they produce the, I mean, a researcher to come and produce the nanomaterials? So I suppose if you have flexibility in that planning, perhaps we can achieve and translate the research into a more commercial, because I don't know, my own way of looking at it, when I mapped into this matrix of integration of existing knowledge for immediate impact in quite a number of projects, I put the projects into that square. That has, I mean, seems to be having the potential. So few things seem to be, I mean, if you look at it that way, instead of looking money, we probably are looking at getting money, because we are changing the position, because otherwise our mindset for the moment is, okay, we need the money, then we'll do this, and then we'll go to the next. I suppose. Yeah. So I think, okay, now we as researchers, it is the economic crisis is the opportunity.
And of course, there is a gap we get in the funding. So what I did, one thing that I understand the dollar was like 200 something, went to suddenly the 300, right? Something like that. That means I understand there's a huge potential for the kind of outsourcing. Outsourcing means not only the work that they can outsource, even the outsourcing of the research. Then what I did was I contacted we have an institute. Through the institute, I contacted many people around the world. So that what we are doing is they send a part of their research and we do it here and send them back. So one area we start the collaboration, UK Institute about the biomimicry, right? We are now, how we are going to apply in the built environment. And there's another one is on construction demolition waste, as I'm doing more work on basically on the built environment. Likewise, we have to be different than before, right? So that as we are not getting the funding from the local sources, we apply more Horizon grant and various other grants. So that likewise, I think otherwise I may not be thinking about getting the research funding as well as research opportunity from the outsourcing, because there are many institutes that we are contacting right now and getting their different kind of job. It's come from the various types. Some of them are like formation of the research for the next research application. Basically, they come up as something like that. Payment for the RA plus or something like that, so that we can do a lot of work. So that actually that opportunity came because of the crisis and this situation. Just for that.
Professor Dissanayake, now your structural health monitoring, you show a lot of pictures now. Bridges are collapsing.
Yeah.
Now, how can we actually do something so that now you said putting up a new bridge versus repairing the old bridge, huge difference.
Yeah.
So on that line, can we do something to the country?
Of course, actually,
Why can't we?
No, actually, we do it. We do it mean simply, we start at research level. So after several times, we establish a company, right? The company actually almost every day 1 or 2 jobs are coming, right? It's coming the different way because now government factories own the assessment before placing the new orders. Every year they have to factory be assessed, but there are certain the structures are there all right. Even in the most of the building that was rehabilitated in Colombo, actually we did the assessment, right? So that they come now, because now in the industry where I am working, like they know that if you want to get the job, that they have to contact me, like so that because then we do it actually. Even from the CJRA side also, I had one discussion. I said that now they have like 850 bridges are in the critical condition. So basically, we have to keep on assisting and doing that. That we of course. We try our best to reach them and get the job and do it, because I have people are working because I want to pay them salary, right? All together, all 5 companies, 320 people are working, right? Now we connected, and so we didn't. I mean, everybody is working, and even recently we increased the salary also because. But we understand the situation. We are not getting the much profit, but of course they are transport and food and everything has increased, so they have to increase it.
My wife was thinking, because if we can show the impact through this project, the impact of the country, if that goes to the treasury, then they might put more money.
Yeah, that is huge actually. When you think about it.
So is there a way of connecting the
Thank you very much for all. Congratulations, first of all, for all researchers. I'm giving my thoughts on behalf of the ministry. As going with the secretary's intention, that's true that all, with due respect to all the efforts of the researchers of this country, or the academics or the intellectuals' efforts, we are at a very critical stage, as you correctly said, on economically, and the social impact will come within another 4 or 5 years to the country back, whatever of the current situation. So being intellectuals, being academics, we have a huge responsibility at this stage, how we have to move forward, what will be the next. So to do that, I would like to pay your attention. If we all get together like intellectuals as well as the ministry and other stakeholders, and I feel like this is a stage, being from the administrative aspect, I feel like as Secretary said, that we too feel like we have a gap. The researchers or the intellectuals brings your outcome through the research or findings, and how these findings take back to the general public. How these findings, how this research outcome can we, how to transfer this technology to the real world. That should be the ultimate goal of our research or whatever our academic efforts. So I feel this is a good forum to discuss that. How can we bridge this gap, your findings, and to the general public? Because this research, especially little bit of, I'm always telling little bit of different from the university research or university, because this institutional research or the applied research, how can we bring them to upgrade the general public or upgrade the economic situation of the country? I know that some of the researchers, as you said, can do research and can make some efforts to commercialize those research through your own companies or own systems, but all academics cannot do that. So if that's the thing, that's the role of the ministry or the other organizational or the institutions in the country or the public sector as well as the private sector or the commercialized, whatever the sectors, that is the place that we have sit together or bridge together. So I feel, I want to say that is our role. I feel that is all of our role at this moment. At the same time, I want to pay your attention, like IBL and UBL system, the National Innovation Agency is currently working on it. UBL system is in universities. So please, that is one of the platform that we can bring together. So all your findings, if we can bring that to that platform, that or one common platform, and through that way, we are trying to bridge the private sector, or we are trying to bring the entrepreneurs to that platform, and will work on it, then your research findings, your outcomes will be there, and we hope it's a kind of matchmaking thing. You will bring your findings to there, and I hope Professor Ajit Dialwis will elaborate on that particular platform which we are planning to bring out in this year. I hope so. Thank you very much once again on behalf of the ministry. Thank you very much for all of you for making this event success, as well as your efforts, your valuable property, intellectual property who brought to this common platform. Thank you very much.
Because we have not on this project, but on other projects, we have gone into commercialization of an anti-cancer capsule with a private company, and then there are other activities and the patent applications and so on. So it had been useful and it is quite active now. So, but the thing is that the structures will have to remain to continue. That is the point I wanted to get across. With at least the minimum to keep it alive, then the people can be active. But if the places are closed, then everything goes for a six. So that's what I want to say.
Maybe request something along the lines that you said. Now there are 15 projects that definitely we saw things are there that one can capture. Now another issue was that what you raised, $1 needs another $100 to commercialize, but that's not going to happen with the current way. So can we reverse it in the direct that, okay, if each of the groups can say, look, we did this, and there is this immediate potential if support is given, and there's a chance, like from the structural health monitoring, like from the amount of spices that are going out in the market, currently going in this way. You mentioned you had aflatoxin coming up topic, but if we can highlight one or two items from each of the research projects and ensuring these collaborations can be there in time, it's not lost, it can continue, something to be done, and then going to a one pay via the ministry, like to enabling some sort of support to keep the projects going and highlighting the ability, potential of the projects. I don't know now, what you mentioned was the aluminium and the green roof. I mean, you had the soap nut, but that can soap nut with the green surfactants come and support on the microencapsulation. I don't know. And then you raised the Z inverters. I mean, okay, it was a kind of like shocking. I was always thinking the 20 years are there now to see that 10 years the inverter is going, right? But maybe the developed countries don't do so much of research because their power system stability is high. We are the people with the instability. So our inverters are very likely to go, but then, okay, the research on that to be very quickly to scaled up and done. So maybe if we can highlight few of those things that have come in and then showcasing, okay, a little bit of money, but keeping the research groups going, there is this potential. Is there a way that all of you can connect? I mean, like, okay, you said Kenya is leading on the pink tea or the new tea, and then you suggested 5 more teas.
There is a potential, and these are already, the husbands are already known. The problem is tiny little effort has to be there to express the key parameters and all, then it's ready to go. It's really almost in the verge of everything is done. And the husbands are maintained nicely. So only these parameters, if they can express and see with some genomic aspect of the environmental changes, timing and when to harvest the potential, when to harvest and all, these things can go to the market.
So Sajid, just for the simple one, actually I'll be opening the GAP headquarters in the Colombo 7 in between ISL and SLAS, so that where the main understanding the gap, so that the whoever want to discuss, come and have a discuss with us. I mean, you can find out the mobile number, and we will, while supporting, if you want any collaboration or finance and various other things, not over the counter, but we can make it, because we have got a big group of the people. Though these are some of the investors, various ways. So we are sometimes try in a big way. Sometimes we go with the smaller way. Some of the work that we are going to, because one is established with a construction demolition company. Another one is manufacturing the bamboo timber, right? Then we are thinking about bringing the investor for the solar, right? We are thinking the Parantan. We think the big and small, no matter which is, but we would like to have a discussion, ilmenite, various things we discussing. But when you discuss 10 things, one might work. As you know that we established the first factory for the fly ash. Now we are exporting to the Maldives, right? Then of course the structure health monitoring, sustainable construction. Likewise, as I said, that the Gap Resource Center, which is the outsourcing company. Likewise, we try in the various ways. So if anybody want to have a discussion, we will, not only for the researchers, we go for the private sector also and the government sector also, likewise, just for your information.
With regard to this solar cell material, Dr. Attegala, is there any chance of having new material suitable for solar cells, more efficient kind of material? Yes. Yes.
So we can continue it in that way, and also think about, using graphene maybe, because we have that, but in a way that we can use for the solar cell devices. So the proposal we submitted to continue the experimental work is with the University of Morocco research team.
I see.
So probably if we get some more funding,
What
we
does
can
the
find
Indian
out.
team think about that work? Is it any?
Yeah, now Indian team actually they did the theoretical part, but we wanted to actually synthesize these materials. So they also joined with the new proposal. Not only them, actually another research group, experimental team also joined. So I think if we get some sort of a support, we can go ahead and.
You are currently still in the experimental stage, but not in the near future that new material can't come. No?
Yes, sir. Now considering what we have done, like our theoretical investigations.
It won't happen tomorrow. I know it takes the 5 years, but if it happens in the 5 years, that's great.
Lots of research are happening, it's not only us. So I think they are using whatever we have published and continuing their research work, in experimental manner. So recently, even in the Princeton University, that research group, they have used two-dimensional coating to, solve some of the issues in perovskite solar cells. So anyway, we hope for the best.
Okay, thank you.
Thank you.
May I suggest, as to Professor Ajit suggested, you have to identify this low-hanging fruit. One way to make sure is to see what are the things which could be commercialized very fast, so that we can impress upon the ministry and the government treasury, put additional funds for research. So my suggestion is NIA can take that role. The final reports are there, and let them go through that with some expert inputs, and to then prioritize shortlist, then to have one-to-one discussion with those parties, and then we identify if it is working, then we can put additional support and go ahead and finish it very soon and come out with something which is basically there is an impact to the country. So we will look at that. I think NIA should take a lead role, go through that, then identify, prioritize, then have one-to-one discussion with the project leaders. That may be because I feel that scientists alone can't do the prioritize very difficult. You need the economic feasibility aspect should come in.
Which includes that we need to have a
dialogue with the private party, our technology can transfer to that sector. We are there to organize whatever the events or discussions or online method or whatever, because we have that technology so easily, not like rather than having sometimes this time of physical meetings, sometimes we can arrange, because ministry can invite, if you say that my product goes to this, my technology can transfer to these potential people. Yes, we are ready to do that. So please, please keep in touch with us as well as the NIA. NIA is the agency. From the ministry side, we can support whatever the writing to them or inviting them about those things we can do. Please come forward and work with us to do something for the to come out from this situation of the country. Thank you.
But can we initiate with the 15 groups coming up with the low hanging, and also to keep the research components going, where they may see other connected areas, and from the getting the ideas and then building on that and then pushing it out via the ministry, so that the researcher should be supported, because there is so much of potential in the what has already happened. So highlighting this potential in the minds of maybe Minister of Finance to keep the support. So okay. Right, then I suppose we have something to do. And the next item, so handing over to Dr. Inoka.
Yeah, thank you, sir. I'd like to thank the panel members for the constructive comments given to the research teams, and also the research teams for their comments. Ladies and gentlemen, it is time to wind up this symposium on Indo-Sri Lanka Collaborative Research Program. Thank you for your active participation and presence at this symposium. You are all invited for lunch at the cafe. Thank you once again, and have a good day.